Good morning, everyone. Welcome to the FlowJo High Parameter Data Analysis course, Chapter 1. Now, before we begin our session, we would like to tell you a few ground rules so that we have a smooth session. Please turn off all your other programs and listen to the speaker attentively. Turn off your camera as it creates a disturbance for the speaker. All of you have been put on mute, and we encourage to ask as many questions as you want by using the chat window, and the questions will be taken care by the speaker at the end. Now, I would like to welcome Ubaldo Barbosa, our National Application Manager, BD Biosciences, who will be the moderator for the course.
Thank you, Ankita, and welcome all of you all for this program on the FlowJo. We are really overwhelmed by your participation and your registrations. The whole concept of this program came up after, we had conducted the previous program where we got overwhelming response when Anjan Ghosh conducted this program. Amlakant Ghosh conducted this program. And looking at the response, we thought we need to get back into this detailed program on the FlowJo. So we have actually made this program as a 3-day program. So today is a day 1 of the program, and then the dates of day 2 and day 3. So we have a good number of registrants for this program. And today we are going to start this program by Timothy Quinn Crawford, very well-known personality in FlowJo. I myself have attended his programs when he used to conduct it online. But the only challenge used to be that the programs used to be late in the night for India. Tim has been very gracious enough to have a late night for him and an early morning for us. And thank you, Tim, for this program. So let me have a quick round of introduction, though Tim doesn't need much introduction. But Tim has been he's been working with BD. He's actually done his PhD in molecular and cellular biology from the University of Washington, Seattle. He was studying development genetics, stem cell biology, and cell signaling mechanism. He gained additional training and experience in HIV biology, viral immunology, and cytometric data analysis as a member of UCSF Core Immunology Labs. Then he joined Hawaii Center for AIDS as a staff scientist, where he worked to evaluate the phenotype and function of the T cells, NK cells, and the monocytes in HIV-infected people. Currently, he's working, as I just mentioned, as an application scientist with BD Life Sciences Informatics, and Tim provides trainings and assistance to scientists all across the globe on single-cell analysis, approaches, and techniques. So today, he's going to take us to the journey of the FlowJo, where he touches upon the basics of the flow in the first part. Then he goes to the compensation, and then we talk about the plugin modules. As you're aware, we'll be having the sessions with breaks. We'll be having, after 1 hour session, we'll have a break where we'll have some question answers session. Quiz will be online. And then after the second hour, again, we'll have a break, and then we close for the day after by 12 noon. Okay, so without taking much more time, over to you, Tim.
Awesome. Thank you, Ubaldo. Appreciate it. Welcome, everyone. Good morning. Thanks for having me. Today, we're going to cover a whole bunch of different topics within the FlowJo platform to enable you to do all of the basic and then the high parameter analysis for your studies. And as Ubaldo said, we're going to start with an intro to FlowJo course that I like to set the groundwork so that you understand all the different areas of the program that we're going to employ for our more complex workflows. And then we'll talk about compensation. Of course, it's important to get your compensation correctly applied, and I'll show you how to perform compensation and some new compensation alternatives or methods that we've enabled within FlowJo. And then we'll talk about plugins to use for high parameter analysis and how to set up those plugins. And so we'll start with an intro that gives you all the basics, and then we'll go into the more high parameter workflows next week as the second part of our course, and we'll be giving out some data and an assignment that you can follow along to understand the workflow. But I'm going to lay the groundwork here today and hopefully give you a really detailed introduction. Don't be afraid to ask questions. I love interacting and answering those questions as we go through the material. And I'll be switching back and forth between a live demonstration using the software and a presentation. So I'll go introduce a concept in a presentation format, and then I'll go show it to you within the application. So I'm going to share my screen here, and we will get started. Just go into presentation mode here. I believe you should all be able to see my screen at this point. I'm going to go into a slideshow. So yeah, my name's Timothy Crawford. Okay, we can see it. Great.
Yes.
And this will be introduction to FlowJo. So my outline here for this first hour is we're going to go through how to navigate the FlowJo version 10 workspace. I'll introduce groups, how you can organize your samples by creating and editing groups in the workspace. I'll introduce you to what keyword attributes are, metadata that you can annotate your samples with and organize your samples using keywords. Then we'll go to the graph window where we'll build up a gating tree and talk about the tools for creating ancestry or gating hierarchies and applying them to groups of samples. Then we'll move to the layout editor, which is where we can format and export graphics and representations of our analysis visually. And then the table editor, where we can make exported statistical lists from the analysis that we have applied. And I'll finish up with a short discussion about how to use workspace templates so you can apply the same gating and have the same visuals and statistical outputs over and over again from week to week if you have multiple runs of data and you want to have a starting point to analyze. And then I'll talk about preferences, which are modifications to the defaults to customize your FlowJo interface and how you interact with the data. So the workspace itself, when you first open up FlowJo, it'll be a blank workspace, so you won't have any of the data or the groupings in here. But the workspace itself is broken up into three main sections. You've got this action toolbar ribbon at the header. It looks like similar to Microsoft Office products these days, where you have a series of tabs, and you can click on these different tabs, the FlowJo File, Edit, Workspace, Tools, and Configure tabs, to get access to different bands of functions. And then you can point to data either as a group or a sample, and then initiate an action by clicking one of these buttons up here. Below the action toolbar is this group and group analysis pane. So groups act like file folders to organize your samples into manageable units. And you can make as many groups as you want. We can make groups dynamic, as I'll show you in a moment. So you can organize your samples in kind of a semi-automated way where it'll pick up the samples based on features within the name or other keyword metadata within the file. And you can use groups to do things like apply master gating trees. So you group your samples and then apply group gates so that they all get the same color and have the same starting point for your analysis. You can also focus on groups within your layout and your table editors to do batching operations and generate reports. Below this group pane is the sample and sample analysis pane. So the samples pane is where you will find all your samples once you load them. You just drag your samples into this pane and they'll load into the workspace. You can also use this Add Samples button located in the Navigate band of actions within the FlowJo tab and navigate to your file folder and bring those samples in. FlowJo loads FCS standard files that you can come off of your cytometer, so you can export those and bring them into FlowJo. And when you first load the files, you'll see a list of the names along with a statistic column that will be empty until we start adding gating to our analysis or any type of other elements here. And then the number of cells column, which simply shows the number of events within the root file. Or when you start adding gating down to specific subpopulations, it will show you the number of events within each gated population. These other columns over on the right, PID, STIM, and well ID, these are keywords. So again, metadata information that we can add here within FlowJo to organize our samples, or we can actually add them when we acquire the data on the instrument, write those keywords into FACSDIVA, and then they're written in the file, and all we have to do is display these columns, and the values are already inserted if you do it on the machine during acquisition. If we focus in on the ribbon, we actually have a number of buttons up here that are actionable. The little FlowJo icon is an application button. You'll get save functions as well as a list of previously used workspaces that you can open. There's an undo and redo button here with the little curly arrows, and then buttons to add samples to your workspace, create groups, the little plus there, calculate compensation, that's our compensation wizard that we'll talk about in the next hour of the program. Open up our table editor to generate statistical reports, and open up the layout editor to generate visual reports. Over on the right, There are a few additional buttons, including this heart icon, which will take you to your preferences, where you can modify the default functionality of the program in many ways, and the question mark, which will take you out to our searchable documentation pages. So if you have a question about any of these action buttons, what to do with them, you can hit the question mark over here, search for that function, and it'll give you a couple pages of how to use that function and what it's for. So the organization is that these tabs group these bands together, and then the bands group similar action buttons. And you will notice that if you start up the application and you mouse over any of these action buttons for a couple seconds, you'll get a drop-down tooltip with a hotkey to initialize the action, so you can actually do things without clicking around in the ribbon if you want to learn those hotkeys. Now, importing data is simple as dragging and dropping those files. When you first open up the workspace, you'll be presented with this blank looking workspace with two groups. All samples and compensation groups will be there by default. You can just drag your folder with the FCS files, or as I said, navigate through the add samples buttons located up here in the ribbon. That'll load your data into memory. If you drag a group, you will also get a group containing your FCS files. The size here in the groups pane is the number of files contained within that group. They'll always go to the all samples group, but you'll also get a group called the name of the file folder if you drag at the level of the folder. So load your samples, and then we can start interacting with them. Again, focusing on the groups pane, I like to start by organizing my samples into manageable units. You can make as many groups as you want, and you'll see you give your group a name, a color, and you can give it an optional role if you want to specify the purpose of that group. Some groups could be for your compensation controls, others could just be test groups, or you can just leave the role blank. And then this size is the number of files contained within each group. When we create groups, you click the plus button up at the top of the workspace toolbar, and that'll open up this create group window where you give your group a name, give it a color, you can choose the type of text that the group name will display as, and a role, as I mentioned. And then there's this box down here called sample inclusion criteria. All you really have to do to create a group is give it a name and press create group, and then you can drag and drop your samples if you would like. I'm just going to go here, drag any of these samples to a different group, and they'll populate that group, and that's manually populating that group with samples. But this sample inclusion criteria box has this button checked for live group, and if you set any criteria in this box, then this live group with the box checked will automatically pick up the samples if they fit the criteria. And so I like to make a couple groups initially by using these drop-down lists for keywords. And again, keywords are metadata information within the file. In this case, I'm choosing the stim keyword, and I'm saying if the stim keyword that I've entered on the instrument and written into the file initially equals this PI plus NS condition nomenclature, then go to these samples. And when I click create group, it'll automatically pick up the samples that fit that criteria that I've set. So you can always create groups, and you can always double click on a group to change the group's preferences or inclusion criteria, name or color, and then apply those changes if you want to update a group. So if you don't like the group you've created, you can always delete it. I often use the file name, which is under the keyword drop-down list as $FIL, because you'll write pieces of information about your samples into the file name, and then you can use that to make these complex groupings. The samples pane, again, lists all these samples. When you click on a group, it will only show the samples within that group down in the samples pane. And as I've already said, these gates can be applied to individual samples or at the group level so that you have universal master gating across a group. The statistics column, these are frequency of parent statistics. When you draw a gate, that's what you get is a frequency of events contained within the child gate from the greater parent population that it hangs off of. And indenting these nodes means that it's a child. So I've drawn a gate for singlets on the root sample, and then within the singlets gated population, drawn a gate for lymphocytes and then for live. And these other quadrant gates off of the CD3 are actually at the same level. So there's a quad gate, that means there's 4 subsets here on the CD3s to define our T cell populations. And so we'll go through all the different gating tools that are available. But this is really useful. You can expand and contract the gating hierarchy using these little triangle buttons to display the statistics in one or more samples and review what the frequencies are once you've drawn a gate. Now, keywords, I've mentioned this a couple times, but these are metadata, pieces of information that we can create within FlowJo, if you'd like, by going to your workspace tab and adding keywords from this keyword drop-down list. There's an add keyword button under there. And you can give your keywords any name that you would like, and that will show up as a column within the samples pane. And then you can enter values associated with these samples for the different keywords. You can also control what columns are displayed here by using your edit columns function, which is available as a right click on the headers of this samples pane, or it's also available within one of the buttons in the toolbar up here. I'm going to go through a few more slides, and then I'm going to load the data, show you how to create a few keywords. I recommend you organizing keywords for your experiment depending on your needs. But once you start using them, you'll never go back, because we can use keywords, as I said, to create groups, but we can also use them for fancy batching operations within the layout editor, and that I'll show you later on. And we'll use keywords in our high parameter analysis to organize samples for a meta-analysis using dimensionality reduction and clustering. So it's really important to understand how to create keywords and what they can be used for. When you're interested in looking at the data, you load it, and then you can double click on one of these samples to get started. You can actually double click on any gate, and it will open up just the events within that gate. If I double click on CD3 positive gating here, I'll get those events contained within that gate in a graph window. And a graph window is a bivariate display plot. Starts out as a pseudo-colored dot plot display, and this window opens up. You've got your events in the graph in the middle, and then up at the top left-hand corner are where the gating tools that we'll use are located. And you can click on a gating tool, a rectangle, a circle, a polygon, and draw a geometrical gate around a population or distribution of events based on the expression of a marker to create a gate, and that again creates a frequency of parent stat. So that's the frequency of events within the child gate from the greater parent population. And then to move into that population, you can double click on that gate or double click on the population in the gating hierarchy of the samples pane in a sample. And that'll move into just those events. The parameter choices you can make by using these drop-down lists. The axes labels are also drop-down lists to choose the different parameters. And as you build up a gating tree, you go deeper and deeper. Draw a gate, double click on it, change the axes, draw another set of gates, double click, change the axes, and you drill down deeper into the samples. Up in the top right-hand corner, we've got navigation buttons. The up-down arrows will actually move you up or down the gating hierarchy if you're in the middle of a hierarchy that you've drawn. So you can move up to the parent or down to the selected child population. And then the left and right arrows are scrolling through the samples list, showing this plot for each sequential sample in the list. So you can go up or down the list by going to the left or right, and I'll show that to you in just a moment. There's a lot of display options that are available in not just the pseudo-color dot plots, and you can turn those display options on by expanding this options bar down here. It expands out, and there's a drop-down type of graph menu. Within that, there are a bunch of different options to visualize these as with different color schemes and gradients. I'll show that on the next slide here. There's also a third parameter heat map option that you turn on by checking this color axis box. Once you apply that, it'll go from whatever view you're in, whatever option or type of graph, to a third parameter heat map. So you're heat mapping a third parameter that you can select from this drop-down list across this two-dimensional space. And we'll be using that color axis box quite a bit to explore the location of cells within dimensionally reduced data spaces that we'll create in our next session. Below the options list, there's an active gate set of options as well, and this is an expandable bar. You select a gate, you'll notice the handles will highlight these little squares at the corners of each of my vertices here. And then once you've selected a gate, these active gate options will become available. You can choose to tint the gate, as I've done here, change the color of the gate. I've done in the preferences, so it's a purple outline. You can increase or decrease the size of the gate uniformly with the plus and minus buttons. You can make a gate magnetic, so it drags the center of the gate to the densest part of the population it surrounds. And you can choose to gate inside, which is the default. That's always checked when you start drawing a gate, so you're including the events inside the gate. But if you uncheck that box, it'll eliminate or remove those events and gate everything outside of the gated region. These are the different display options that you can choose from your options drop-down menu. Pseudo color is the default. It gives you density information as well as two parameters of distribution. Just to go into more detail here, we're looking at these dot plots with pseudo color. Each dot is one or more events at that position within the graph, and then the density of events is color coded. So red is very high density, and it goes down the contour to yellow, green, light blue, and dark blue to the outliers. You can get very similar information using a contour plot or a density plot with outliers showing here. Zebra plots, some people really like them. It doesn't truly show density, but it's it's a similar type of representation. And then there's a monochromatic option, which is just a black and white dot plot. You can actually choose to make these color dots red or blue or whatever, and make overlays of dot plots that represent different populations within the same data space. But the default dot plot is nice for black and white publications. I personally prefer contours or density because it actually gives you more information, but dot plots are still used quite a bit. The color map option, this is me turning on that heat map option and looking at the PE signal, which is perforin, CD3 versus HLA-DR. These double negative population events are going to be mostly natural killer cells that have very high perforin levels, and so we see more red-shifted colors where there is a high median intensity of those events at that position within the graph. And again, we'll use this color mapping to look at the location of expression in dimensionally reduced plots in the next session. And of course, we do have our univariate histogram, which does have its purposes to look at phenotypes and for gating, and we'll use those over and over again for various purposes in the next couple sessions. Now, the gating tools themselves, there's a lot of different gating tools in FlowJo. I personally love my polygon. You can just go in and click on every time you click within the graph window here, it'll create a new side. Double click closes the polygon. Rectangles and ellipses, you start at one corner or one end. Corner of the rectangle, hold down the mouse button and drag it across the space that you want to include. That'll open up the rectangle. Ellipse is the same way. You hold down the mouse button, drag it across. And this contour auto plot tool, it'll follow the contour line of a population if you want to gate it really close, which is kind of nice. And you can move your cursor wider or closer towards the center, and it'll make the contours that gate tighter or looser around those populations. And so again, we're going to start by gating. Oh yeah, please go ahead.
Tim, I don't know whether you would like to take questions now, or should we keep the questions for the end of the session?
Well,
or,
toss me a question
break?
now. If they're throwing them out, let's address them.
Okay. So Sandeep, you've raised your hand. Do you have a question? Quinn, can you quickly answer that question? Ask that question. Okay, Karthik, your hand is also raised. Okay, so I don't think they want to ask. Okay, so Tim, you can carry on. I think they can.
Option, guys, is just type in your question, and then Amlan can go ahead and ask it for you.
Sure.
No worries if you.
Yeah. Okay, Tim, you can carry on. Yeah.
Okay, so I'm going to load some data now. We're going to build up a gating hierarchy, and this is kind of where we're going. We're going to start with a singlets gate. We're going to build up a gating hierarchy. I'm going to show you how to add statistics. I'm going to show you how to do Boolean combination gates from single markers. And remember that these statistics that we're going to get are going to eventually be pushed out in the form of a text file so that you can work with them downstream using our table editor. But first, we need to create the gating, creating the analysis on a single sample, and then we'll apply these gates to a group of samples using our groupings. I'm going to start that, and we'll talk about transformation in just a few minutes. So I'm opening a new workspace here with our FlowJo application. So here is V10. We'll spin it up. The current release is 10.7.1. If you're on an older release, I do encourage you to just update when it comes out, because you always get new tools and a better performing program with those updates. And so here is our blank workspace that we're working with, and you can resize the workspace, you can change the different areas of it, and as I mentioned, you have these different tabs that you can click on to access the different bands of functions within FlowJo. And when you first start clicking around, you might notice that a lot of the action buttons are grayed out. That's because we don't have any data, and a lot of the functions require you to point to a sample file or a gated population to use them. And so standardly, when you don't have the right selection to perform an action button, it will gray out that action so you can't select it. The moment you click on a sample, those will become available. So I'm going to bring in my data simply by dragging and dropping. There's my pfix data. This group gets created. My data files load into memory here and are ordered by the file name in alphabetical order initially. Now this group is completely modular, so I can delete the group without deleting the samples from that group. Notice how the group is gone. I'm just pressing the delete button, saying, okay, I want it to go away, but those samples still live in the all samples group. I show you this because the only way to remove a sample, one or more samples from the workspace, is to go to your all samples group, select that group, then select one or more samples and delete them. And if you do that, the sample will actually be removed from the group. It will be removed from the workspace if you're on all samples. If you accidentally do that, it's okay. You just bring in your sample again. Here's my compensation control. Little windows at the bottom right is giving me problems, but now here's my compensation control that I just removed, and if I drag it back in, there I'm back to 46 samples. So you can't really go wrong there. But I'd never like to gate on the all samples group or use that group for all of my analysis. I'll mention why in just a few minutes. Instead, I like to create at least one additional group, and maybe I will call this group gates or gating group or something like that, master gates. In this case, I have a bunch of different types of samples here. I've got compensation controls, and you may notice that the compensation control group automatically picked up those 12 compensation controls for this experiment. That's because these compensation controls have the word comps in them. And if you double click on the compensation group, it'll show you the group properties here, and the compensation default group has sample inclusion criteria already set up. It's a live group to pull in samples based on the criteria that I set. And what this criteria says is if the file name contains the word unstained or the file name contains the word comp, then go to this group. And so because I've named in my specimens, when I set up my experiment on Diva, bead comps or cell comps groups, those file names are there, and it picks up the word comp and it automatically puts those samples into my group. So these other samples, I've got FMOs, which are fluorescence -1 gating controls, leaving one antibody out of a master mix to show where the true negative is for each individual marker. And then I've got my real experimental samples that are all my LD samples here. LD1 is the patient, LD2, LD4, and then I've got the stimulation condition included in the nomenclature for NSNS, NSPI, PINS, and PIPI. That's different lengths of stim with PMA and inomycin across these samples. And so I've got 5 different patients in this example, and each patient has been stimmed under 4 different conditions. So what am I going to do with these samples? I'm going to organize them into manageable units by using my group button, creating a group here. I can give my first group I'll make my all stain group. So my all stain is what I call my real experiment, all of the LD samples in my experiment. I can choose a different color if I don't like that one, choose a text style, that'll be the text style of the name of the group in the groups pane here. I'll make it bold italic, and I can say that this is actually my test group, my real group. Now, if you have multiple panels of reagents, like you do a B cell panel and a T cell panel, you'll see multiple lines of reagents up here if you have all of those annotated. And you can just click on one of these lines and say create group, and all the samples that are annotated with the same set of reagents will go into your group. I only have one panel here, right? I have different stimulations and conditions, but I only have one staining panel. And so I'm not going to use that staining panel as the inclusion criteria. That would just bring all of my samples in to the same group, and I already have them here in all samples. Instead, I'll use this keyword drop-down list and use $FIL. You may need to search for it the first time you use it. $signs are keywords required by the FCS standard. We've got a whole bunch of information in here, the date, the beginning time of analysis, the cytometry that you acquired the data on, right? All of this information is metadata contained within the file. And again, we can display this information, we can export this information, we can access this information. So I'm going to use the $FIL, the file name, which has all of this information in it. And I'm going to say from the next drop-down list, if these files contain the acronym LD for lab donor, then go to this group. Now I'm going to create that group, and notice what happened. I get an all stain group. It picks up all of the samples that have LD in the name, and now I have that group as a unit, and I can click on it and limit my view to just the real experiment. I'm going to leave this open and say I'm going to do an FMOs group and go ahead and let's say this FMOs group is going to be a control group because it is a set of controls. And instead of having the file name include the LD name nomenclature, I'll use the FMO acronym. Create that group, and I get a group called FMOs. Now, my last group, I want to merge these two sets, right? I want to take all my all stains and my fluorescence -1 controls and put them into a single group. So I'm going to call this my master gating group. And I don't like this color, so I'm going to give it a nice bright red. Call this my gating level group. And instead of just having the FMOs come into my group here, I'm going to add an additional layer of choices by clicking more choices. And I will use this next drop-down list to say, or if the file name contains LD. So you see how this is building up. I have a file name if it contains FMO, or if the file name contains LD. I want all those samples to go to my group called master gates. Create group. There they are, my FMOs and my LDs. And so notice now I can scroll through these groups. I'm going to close this window. And I am limited to just seeing the samples contained within those groups. I can always get to my all samples group by clicking the top level, or I can go to any of my individual groups by clicking. And we can use these groups to apply master gating, or we can use these groups for other purposes, like batching in the layout editor or iterating over the samples, a limited set of samples with our table editor to generate our statistical report. So now I have my groups. I want to go down here and I might want to organize my samples further. Maybe you only have specimen 1 through specimen 20, right, shown as your names. Well, you can always add keywords to describe your samples as individual columns. And as I mentioned, I like to add keyword information on the instrument. So there's a way you can do that, similar to adding the labels for your antigens that are targeted by each color, representing each of your different channels. You can do that on the instrument. And then when I come into FlowJo here, I need to actually then I can actually display those keywords, and there's two ways we can do this. So configure tab here at the end gives me an edit columns function, or I can right click on the header of a column here within the samples pane, and I have that edit columns function available as well. So either way, I get to this edit columns graphical user interface. On the left are all of the potential values, the headers of the keywords in the files. Things, you know, there's a whole bunch of information written into these files, not just the data. And what we do here is pick a group of keywords, headers, to display over here, and we move them from the left to the right by double clicking. So maybe I do want to show which date or cytometer, let's say, the samples were collected on. So I can move that $site over by double clicking. These reagent columns are the targets of the antibody in that color. And so I'll show you PAC blue and PE Texas red by double clicking. You can also multi-select and click add column here to move these over to the right. And then I'll show you just a couple of my user entered keywords. So this is patient ID and stim condition. And then finally, let's go ahead and do well ID, since this was a plate-based assay. And so again, this is all metadata information. And now when I click OK, I will see these columns available to me with the information included in those columns. So when we look at a sample here and we look at the PAC blue reagent, right, the PAC blue parameter, it'll say CD8 because we've already labeled that PAC blue, that channel represents the CD8 staining. PE Texas red will represent CD4. You know, the cytometer is just showing what machine I acquired on. And these columns, you can go into them and actually change the information in many of them and most of them if you display them. So let's go ahead and open up a graph window here. Double click on this first sample and you notice it opens up the data over here on the right. If I click on the X or Y axes labels, I'll see a drop down menu with the different options for selecting the parameters that we've acquired in this experiment. And here down at the bottom, I've got my compensated PAC blue area parameter, and that represents CD8. It shows CD8 there. The reason this shows CD8 is because I actually labeled that on DIVA when I acquired the data, so it's already written in. If I had mislabeled that and it was something else, it was a different marker, I could display this reagent column using my edit columns function, and then go in and change the number or the designation here. Maybe I mislabeled this and it's actually CD45RA, right? So I could type that once. Do I need to type it 46 times? No, I don't. I need to type it once, and then we have a function within the keywords band, which is under your workspace tab, to copy that value to the group. So I select the value that I just typed and copy the value to the group, and it applies that value that I've just selected down. And notice what just happened is that the comp blue area parameter that this represents is relabeled with my CD45RA designation. So I'm showing this to you because a lot of people don't label on the front end. And so if you come in and all you have are your parameters within these drop-down lists and no mention of the antigen that you're targeting with your antibody fluorochrome conjugate, then you can display that column, type in once, highlight, and then copy the value to the group. I'll just do that once more. I'm going to go CD8 because this is actually CD8, press return, go back to that cell and copy that value to the group, and it updates to CD8. Once you've displayed these columns, you can always go back to edit columns, remove them from display, remove columns, and then press OK, and notice the columns have now changed. I usually like to leave pertinent information up and may remove the information if I'm just using it to label a antigen. So that's your basic showing keywords, and that you can actually create these lists. But maybe I need to add my own keywords. So I haven't labeled anything on the front end, and maybe all of these are sample names that don't mean anything. Well, I can always add a new keyword, and I'll actually do this. Doesn't matter which one, but I'm just going to go to my list of 20 real samples here. I'm going to add a keyword. I'm going to call it my keyword. Right? If I create a keyword, give it a name, I get a blank column, and now I can type in information. This can be alphanumeric up to 64 characters. I could go 1, 2, 3, 4 following the stems, or I could do 1, 1, 1, 1 following the patients, right? The next one could be 2, 2, 2, 2. Or I can give it any alphanumeric label that I want. If you do want to create a list of keywords, there is a function to create a keyword value series here, which can either be a numerical set of numbers, right, a numerical series, or you can set up your list of keywords here, type them in once, and then repeat them over a certain in a certain form using the fill, value, and start over. I'll just show you with the numerical one. If I start with a numeric number of 1 and I want to make a list of 1 to 20, well, that's the default. I can just press OK. If I want to reuse the first number 1 4 times before starting over, then I use the value 4, 1, 1, 1, 1, 2, 2, 2, 2, 3, 3, 3, 3, which again follows the LD nomenclature, right? That it would be, these would be 1, 2, patient 3, 4, 5. And all you have to do is click OK. If you want to use those numbers one value at a time, but start over every four samples, then that gives you a numerical series of 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. And the same thing can be done with numerical or I'm sorry, alphanumeric keywords by using the keywords tab here and filling in the keywords once and then iterating over a series of samples, because oftentimes we have a repetitive organization of our samples in terms of the patients and the stimulations or the conditions that they were stained under. So I'll just click OK here, and you notice that it fills in with that numerical value. Keywords, again, are going to be really useful downstream. Not only can we export these keywords to describe the samples when we export statistical tables, but we can also use keywords to do batch iteration within the layout editor, which I'll show you shortly. Okay, so that's bringing data in. Let's go ahead and make some gates. When I start out, I like to do forward scatter area versus height on as a starting point, or sometimes I'll actually use time versus area. But this forward scatter area versus height, here I'll transpose it once or twice, doesn't matter which way you look at it, is a mechanism of removing doublets from your samples. So I always recommend that you at least turn on the height pulse parameter and acquire that when you're forward scatter, and you can also acquire width as a pulse and do doublet discrimination there. What I was always taught is to use area versus height and then take these events on the diagonal that are going to be our single cells and remove the cells that are off the diagonal. So I'm going to use my polygon tool here, and I can just draw a nice little gated region, double click to close the gate, and call this gate single cells or singlets. So notice I typed in the name, pressed return, it closed the gate, gave me a frequency of parent statistic on my gating, on my graph window, and now I can actually work with that gate. Notice that the numbers are fairly small, right? And I'm using black and white. Well, these are preference changes. I often like to go to my preferences, my heart icon, and make modifications to the defaults here. And I'll show that to you right now so you know where you can do it. One modification, these are different areas of the program where you can modify the default functionality, and maybe I want to make this a different color gate or increase the line width or the increase the size of the labels here. Well, under gates preferences in the top line, I can choose, well, what's going to be my default line weight of my gating? What's my default tool? What's the default color, right? So the tint color will be my default gate color. I'm going to make that purple. One of the favorite things that I like to do is you may notice that when you first start drawing gates that your label is right on top of the population. Well, if you open up your gates preferences and go to annotation position here, you can choose to change that to being from inside to above or below or to the left or the right. So I always just choose above. And then when I draw a gate, that label ends up at the top of the window above the gate, not right on the population of interest. So now that I've changed the tint color and made the gates darker, let's go ahead and double click on this population and move down into that subset. It opens up a new graph window with just those gated events in it by double clicking on the gated region. I can also open up this gated region over here within the gating hierarchy that's being created. Double click, opens up those events. And then I can change the parameter that we're looking at. So maybe you want to go to side scatter now and do a kind of lymphocyte gate. And the lymphocyte gate, I'll go ahead and just use a ellipse to get the major population of cells. These are going to be my monocytes. These were Phi-called samples, so the grands are gone. But an ellipse, you hold down the mouse button and drag across, as I said, the population. It opens up that ellipse, and you release the mouse button. It brings up the window to name this, and I'll call these lymphocytes. Yeah, lymphocytes. And you can always resize and change the gate here. So I move the gate around. Notice that the number updates automatically depending on where I drop the gate. These little handles, the square handles, can be grabbed to resize and change the gate positioning. And then when you're happy with that gate positioning, again, you can double click. So I noticed something again. I only changed my dark purple gating color, not the actual name here. So I'll go to my preferences again, and I'll just go to my fonts preferences here. Fonts allow me to change some of the default colors and sizes of the fonts in different areas of the program. And if you mouse over any of these, it'll give you a tooltip saying exactly what you're changing. In this case, I want to change the statistics on the graphs. That's the actual labels here. And maybe I'll make them bigger so you can see them. 18 point, and maybe I'll match the gate color with purple. Okay, so notice how that updates, and now I'm seeing larger font. You can read it. If you're sharing this with a boss or a colleague, you know, it's nice to be able to actually read the gating frequencies. And so this is one method that you can just increase the size of those. I'll go ahead and double click on this lymphocytes gate, and we'll go into those cells. Let's see, my next gate I might make is a live dead staining gate. So dead cells are stained with this aqua-amine reactive dye in this assay, and they end up being very bright, taking up the dye, and it binds all free protein. So these negative cells are actually coated with the dye, but they're distinctly lower in intensity. And so with this live dead discrimination, we're going to keep the live cells with low ARD expression. And I'll use my rectangle gate again. And now that I've made those preferences changes, you can see that my gating color is maintained based on the preferences I chose. Now, actually, when I'm doing this gate, I use a modifier with my polygon tool. Without any other buttons held down, I can go at any angle relative to the last arm of the rectangle or the polygon. If you hold down the shift key, it'll actually keep you at 45-degree increments relative to the last line. So you can make nice straight lines by holding down shift. Double click, closes, and I'll give these a live cells designation. All right, and again, once you've created that gate, you can always resize it, change it by grabbing the handles. And when you're happy with the gate, double click to move down into that population. I will look at some more interesting markers now, HLA-DR on the Y versus CD3 on the X, and you've seen this plot before, right? It's that CD3 T cells, these are going to be HLA-DR positive, mostly B cells, and double negatives are going to be my NK populations. And here now I can isolate the CD3 positive T cell population using my favorite tool, a polygon. So holding down the shift button, I can cut right through those two populations. Without shift, I can make an angled gate at any angle relative to the last one so that I can move away from that spreading population of B cells, and then hold down my shift button to close the gate up and make nice straight lines and call this CD3 positive. Now I'm showing you really nice data, right? This is data that is well distributed. Sometimes your data looks really nice coming off the instrument. Sometimes it may look squished around the zero axis, around the negatives might look squished, or they might look really wide or spread out. And these are visual changes that you can. This is due to the binning of the data within this visual space that's being presented. We're actually looking at data by default on a bi-exponential transform function, and bi-x means that there's a linear range around 0 that displays all the low-end events, that you can see the negatives and any negative events within this plot. And then there's a log range further out. So a linear range around 0, and then it turns into a log scale so we can see the 10 or 100 or 1,000 or 10,000-fold difference between a negative population and a population that expresses the antigen of interest. So this scaling here, the bi-exponential scaling, they can be changed and modified to make your data look very different depending on your needs. And it's just a visualization change. It's not actually changing the raw data. The way we can change the data is by going to this T button, T for transform, and we can go to a straight log axis or a linear axis, or to customize axis, which is where I would go to make modifications to a bi-x transform. Because log axis, if we do it on these data, well, I'm missing now half of my negatives. Any events that are actually below 0 on a pure log scale won't be displayed. And so this is 1/2 of my negatives, and it gives me a warning. 9% of events are off the chart edges. That's everything that was below the visible range in this pure log scale. A linear axis change, that'll squish everything initially. You can still change the range and pull it all out, but it won't be appropriate for most immunological markers. In the case of certain dyes, you do want to use a linear axis if you're doing proliferation or cell cycle analysis. Then you would want to use a linear axis for those parameters. But in most of our standard antibody staining, we're going to be looking on a log scale of 100 or 1,000-fold difference between negative and positive. So therefore, you do want to see data, the positives on a log scale. And what we use that bi-x for, that as a standard, is allowing us to visualize the negatives and the positives all within the same plot. So to go back to my bi-x, I'm going to my customize axis window, and I'm going to choose the scale drop down here. What this does is open up this parameter in a histogram view, and you're looking at all events in a histogram. You can see that some are stacked on the 0 point here. So I can use this drop down scale option to choose a different type of transformation. With most of our flow cytometry data, we're going to be using the bi-exponential transform function. With a bi-x, you have the option to increase or decrease the range of this parameter by clicking these plus or minus buttons or just typing in the number here. The default range on our BD instruments is 262144. That's the maximum. But maybe I want to reduce that because I want to spread out that distribution visually, so I can take away some of this high-end white space by clicking the minus button, right? And that's what I've done already to make this data look really nice. Effectively zooming in on that histogram or that distribution. You can add extra negative space to your data by grabbing and moving this extra negative decade slider. And that essentially adds additional space on the back end, right? So move all the data over if it's on the squished on the x-axis. Excuse me. Then I have this width basis and positive decades function. The width basis controls how much space is given to this linear range. So with a right motion of the width basis, notice the compression of the data around 0. If I go to the left, it spreads the data out around 0. And so sometimes your data looks really wide and spread out. You may want to go and make your width basis to the right to a larger negative number to compress, and then you can take away the extra negative if that doesn't look right, right? Or you can set the positive decades, and it does kind of the opposite thing. Positive decades, if you go to the right, it will spread out around 0, and if you go to the left, it will compress. And what this is doing is saying how many log range decades are we going to display here. And so most of the time off the instruments, you'll get a default of -100 for your width basis. Often that looks a little too squished for me, and if you want to look at dim antigens, you might want to spread it out. But for most cases, it'll look really nice, right? I'm just saying that you have the option to change these parameters or these scaling factors, the transformation of your data to make your data better. Because, you know, if you take away all the extra negative space, spread out your data too much, and then apply that type of transformation to the two parameters here, the DR and the CD3 parameters, I'm highlighting both of them and clicking apply, what happens? Well, 25% of my data is off scale. I didn't give it enough space in the back end of this transform function to get everything on scale. Going the other direction of extremes, you can go and compress way too much, add too much extra space, and apply. And now what are you doing? Wasting a whole bunch of space that you could use to spread out your distribution in a nice way. So there's a sweet spot. And you know, this recommendation comes from the masters of flow cytometry in the field. Over the years, the consensus has been to use these bi-X transforms for most flow cytometry data, and that you can transform and scale the data as you wish using this transform button. As long as you transform all the data you're comparing in the same way, it doesn't matter which transform or scaling that you use. So the display is actually up to you, but there is a nice way to present your data, and there is a really ugly way to present your data. Using the T button, you can get both. So I'm going to jump back here and just mention that. I already said we've transformed the data, and one thing you can do to make the scaling look different than the default is going to your T button, customizing the axis, and using the range buttons up here at the top end, the extra negative decade slider, the width basis slider, and the positive decade slider to change your transformations and the scaling for that visual display. So when you apply these, remember that you can choose more than one parameter to apply. So I could select all my fluorescent parameters, click apply, and these settings will be applied to all of those parameters, or you can do one at a time to customize the visualization of a single distribution at a time. If you would like to save the choices that you've made here as the default for this parameter from this instrument in the future, then you can click the Save button down in the bottom left in this window, and that will save these choices. The next time you bring in a file from the same instrument with the same labels here, you'll get the same scaling that you set. So that's how you can save all of the transforms to your preferences, to your liking, as you make changes in your transform of your own data. Now, when you set these transforms, I already showed you this, but you can set them, make them, the visual displays of these events look very different. And I in that now and get choppy, but so these are recommended distributions, right, that I would display, even though they're still valid in their own right. I would choose to spread the data out visually so you can see what's going on, and this gets rid of the squishing of cells around 0, ensures that you see the visual median that is consistent with the statistical median, and makes the plots look better to the eye. So if you're going to share this in lab meeting or in a publication, try your T button in making the data look better. Go into my next slide here. There we go. Okay, so we're building up a gating tree, and now we're going to group apply that gating tree. So I'm going to draw a couple more gates, and then I want to show you how we can actually take that gray gating tree, which is only applied to a single sample so far in the gates that we've created, and drag and drop, or you can actually right click and copy to group, copy analysis to group, and that will apply the gating to the group that you drag to or the group that you apply to that you have highlighted. This motion of drag and drop or apply to group function will turn those gates red. The group gates will apply, and every sample that's within that sample, that group will get the same initial starting gates, and they will all fill in. And so this gives you a mechanism to have a universal master gating applied to a set of samples to be consistent across your experiment. Once you have group applied gates, right, my group applied gates have been applied, you will notice that you can no longer delete a single gate from the actual sample gating tree. If you want to delete group-owned gates, you have to go to the group gating tree. When you do delete a group gate, it'll give you a couple options. Do you want to delete the gate entirely? That means that this gating in the group and on the samples is gone. You'll have to recreate it. Or you can allocate to either the first sample or to all samples. So that means that the gating will be removed from the group, but it'll remain as gray gates on a single sample or all samples within the group, or you can just cancel out this operation. But it's really important that once you create a group gate to know that you can only delete it from the actual group that it's gated on, not within a single sample. The second thing I want to point out is once you've applied group-owned gates here, they've turned the group color, you have your master gating applied. Well, if you make changes to a single sample, you can move a gate and it'll change the color back to gray, denoting that it's actually only that change has been made to a single sample. If you want all the gates in your group to stay synchronized, so if you move one gate in a single sample, it'll automatically apply that change to the group. You can check the synchronized box, and you do that by double clicking on the group and checking the synchronized box. And then you can go into a sample, move the gates around, and they don't turn gray, they automatically apply to the group property, right? So check the synchronized box, click apply changes, and then close, and then you'll be able to move gates on any sample, and this group will stay synchronized, and you will have the exact same gates on every sample within that group. We will get to additional statistics. I'm just going to go over it here, but then I'm going to show it to you. Statistics such as medians, geometric means and means, standard deviations or CVs, these are all descriptive statistics that you can add in addition to your standard gating. And I like to do this through the Add Statistic button, which is under the Workspace tab. It opens up this graphical user interface where you choose your statistic, and then you choose the parameter that you are going to apply that statistic for, right? So I want the median of the Alexa 488 channel. Clicking OK or Add, then it applies a new node called a statistic node that has a different icon here, and it says the type of statistic that's been applied, the channel, and then over on the right in the column, it'll show the actual median fluorescence value that for those events within that channel. And so statistics can be descriptive in addition to, it can describe your experiment in addition to just frequency measurements that you get out of gating. It is a measurement of central tendency, a median, that is a way that we can describe the average intensity of those events within a population for a given parameter. And so we'll use descriptive statistics like medians in our high parameter workflow to describe the populations we get out of clustering algorithms. And so I want to make sure that you know how to add medians here so we can go through a workflow next week utilizing them. Boolean combination gates are a way to create combo gates to cover all potential phenotypes if you have multiple markers that you're looking at. So in this experiment, I'm actually gating down to T cells, and I have 3 functional markers: interferon gamma, perforin, and phosphorylation of the ERK extracellular regulated kinase module. So there's 3 different downstream functional markers that I'm gating. And if I want to create gates of all plus minus combinations for those 3 markers, well, I can use create combination gates under the tools tab. And the mechanism here is to gate on your single positive markers to generate those gates. Then you select those 3 individually gated antigen positive gates and use the create combination gates button, which is under the tools tab under your Boolean band. This will bring up this window where you can abbreviate the names of your gating that you gave originally. In this case, I'm just removing the plus signs from each of them because those will be added during the combo gate creation. And then when I click OK, that'll first create the not gates, so not interferon, not perforin, not ERK, and then all of the and combinations, so triple positives, double positives, single positives, and triple negatives. It's always going to be 2 to the n number of combinations. of starting markers. So 2 to the third gives me 8 potential combinations. But if I start with 4 markers, I'll get 16 gates. 5 markers, 32, right? So this scales up quite a bit, but it's a way to look at all combinatorial possibilities to get at the potential phenotypes or combinatorial phenotypes using single marker gates as your starting point and then creating the Boolean combination gates as an output for all combinations. So let's go ahead and jump in here and draw a couple more gates here. I'm just going to double click on my CD3s and look at 4 versus 8. I always look to 4s on the X. Versus 8, this is a perfect distribution for a quadrant gating tool. With a quadrant gate, you can click on the quad, set the gates here. Notice I've just created 4 quadrant gates with the frequencies there, and then I'm going to double click on the Q1 CD8 population, and I'll just gate my individual markers. Here's interferon gamma. This is an unstimulated sample, so you don't see much gamma yet, but we'll see it once we start applying these gates to the group. So I'll just make a gate here called IFMG. And I can always resize that or move it. I'll go to my perforin marker next. That's the perforin expression within the CD8s. Again, define the positive fraction called perf positive. And then I'll look at my phospho-ERK marker. Right? So this is phosphorylation of the extracellular signal regulated kinase, and we'll see this go up in our stimulated conditions as well. p-ERK positive. So there's my 3 markers. I notice that there's a lot of white space when I'm gating here, and I can probably spread this distribution out better by going to my Customize Axis T button, removing the extra negative decade space that I have in there, and then I don't need to do that 3 times. As I said, you can select multiple markers and scale them the same way just by highlighting them. Click Apply. Now I have more visual spread. The distribution is better suited for seeing these events move into my gate upon stimulation, and I've removed all that extra white space. So again, you can do this step by step using the T button to customize your axis and improve your visualizations. Now that I have those gates drawn, I want to go ahead and make the combo gates out of them, and I'll show this to you on a single sample, and then we'll go ahead and do the group application of gates. So each of those individual markers, actually I'll use a hotkey, Shift Command W here to close all of my windows in one shot, just get rid of them instead of clicking them all. And now I've got this set of gates here that are individual markers on my CDA, and I want to do a combination mechanism using my Tools tab, Create Combination Gates option to create all potential plus minus combinations of these 3 markers. Again, I'm going to remove the pluses, because those will be added during the AND gate or combo gate creation. Click OK, and I get all of those gates, these AND gates that are created, show me all the potential phenotypic combinations of those 3 markers. Now to apply these, I can see that there's not many events that are triple positive. In the unstimulated condition, only 42 events fall into that positive population. But let's go ahead and look at a stimulated condition. So to do this, I'm going to go to my master gates group first, and I'll show you 2 ways to apply gates to a group. One is to highlight all of the gating and drag it and drop it to that sample or to that group. I'm not going to do that because I want to show you the other option. Highlight all of the gates, all the gray gates that you want to apply. Make sure to highlight the group first in this case. Right click, and you have an option to copy analysis to the group. So I like to use that. I've gotten really used to using the right-click copy analysis to group. Just remember that if you're going to do that, you need to highlight the group first, or else you send your gates to the wrong group. When I click on that, it now sends those gates to the group. They become red group-owned gates. It always turns the color of the gating group that you applied. And now I see all of the numbers fill in for all my samples. And so I'm going to collapse all these samples, go back to my LD1, look at that triple positive, 42 events, 0.12%. Well, what happens when we stimulate for 2 hours? After 2 hours of stimulation, I've got 27% of my events, 12,000 downstream, and I can always open that up, go downstream, look at different markers. Maybe I want to do CD38 versus HLA-DR and get an activation state for that, right? I can always add additional gates downstream. So again, if you don't like your gates, you can always delete them. If they're gray gates, you can delete them from the individual samples, but if it's a group-owned gate and I press delete, well, it's going to give me this warning saying I can't delete a group-owned gate. I have to go back to the original sample. So if I want to delete any of these gates now that they're group owned, I need to go up here and delete them from the actual group. Pressing delete here again, I can delete entirely, or I can allocate to first sample or all samples, or cancel the operation. I'm going to do all samples just to show you what happens. I've just removed the group ownership of the lymphocytes gate down, but it remains, these gray gates remain on all samples. That's what allocating to all samples does, right? Removing group ownership, keeping the gates. And if I want to, I can always push these back to the group. I can even drag and drop them like that to reapply, right? So I've got this group own mechanism. If I go in here and I start making changes to any of the sample gating, you'll notice that you can always move a gate once it's group applied, right? I can move a gate here, the live node changes, and it grays out that particular node, showing that it's different from all the other red gates. At this point, if I want to apply that change on a single sample to the group, I'm going to have to override the existing live gate, so I can drag it to the parent up here, and now I've just overridden that. They've all turned red again. But if you want these to be all connected, as I said, you can go to the group pane here, and you can check the synchronize button, click apply changes. Once that synchronized button is set, then when I actually do any gate movements on this sample here, and if I move the live gate, it's not going to gray out anymore. It's just going to update across all the samples. So it's just automatically applying that change, and all the downstream statistics are updating. And so a lot of the time, what I like to do is gate on a single sample, group apply, and then go through and review the gates, but using synchronize so that when I make a change on one sample, it automatically keeps that change on every other sample, keeping the analysis consistent across all the samples in this experiment.
Tim, I
Yes.
think let's have a short break. I think we have our business director would like to address the group.
Sounds good.
Yeah.
I'll just,
So
yeah,
sure. Sorry to interrupt, but then he'll have a Puneet Talwar is a business director of BD Biosciences India and South Asia, and he would like to address the group. This is something which is an education program which we are continuously doing it, and Puneet Talwar takes our director takes a personal interest in interacting with the team members. So over to you, Puneet.
Hey, thank you, Peter, and thank you, Tim. Peter, I didn't want you to interrupt actually Tim during his presentation, but I'm sorry, Tim, if we just buzzed into your presentation. I'll not take much of your time.
Thank you.
I think the program is going on beautifully, and I think there's a lot of interest and a lot of interesting questions coming up. So I would like to let that continue. Just for the participants, I just wanted to welcome you to this meeting and assure you that we at BD are always trying our best to bring out all the latest solutions which enable you in your research, may it be instrumentation, reagents, may it be application support, or the software solutions that we really pride ourselves for. I hope this program will help you learn the latest things. And still, if you would like us to improve somewhere, or where you feel that we have some shortcomings, or where we can provide you even better solutions, please feel free to give your feedback, because it's your feedback which makes us improve every day, and that's the motto of our company and our life. So with this, I would not like to interrupt more this program. I wish all of you happy learning, and thank you, Peter, and over to you, Tim. Take care.
Thank,
Bye-bye.
thank you, Puneet. Thanks for your kind words. Tim, do we have a break, or we continue the session of gating, and then we have a break for the participants?
Yeah, I think we could do either. Would you like to go ahead and do a quick break right now, a 5-minute, and then I'll continue, because I'm about to transition into the layout and table editors and finish this up, and then we'll go into compensation right after. So
okay,
we can just why don't we take the 5-minute now?
Sure,
it's a good point.
I think we are done with the gating, and then we go into the layout edit and table edit in the next session. Okay, thank you so much, Tim.
My pleasure. Yeah, thank you guys for all your attention.
Yeah.
Are there any questions that I could answer or anything like that? Is there?
Yeah, Tim, there are a lot of questions, so I think I would give it to Amlan. He will read out the questions for you.
Sure. Yeah, everybody can take a break, but use the restroom, but if you want it, we can go ahead and continue to talk, right?
Sure, we can do
Yes,
that.
hi. Hi, Tim. Hi, Peter. Good morning. Great
Good
session so
morning.
far. So we have a bunch of questions already lined up, and maybe I can just read out few of them for you. So
Mhm.
first question comes from Karthik, who wants to know whether color parameter needs dot plot overlay, or we can do without overlay.
It does not need, it can be used in the graph window for any set of parameters. You don't need to overlay anything. The overlays create a color-coded system for comparing samples that I'll show you. But no, the color axis can be used in conjunction with any two parameters, and it is a separate option of visual type of graph that we have. So you can turn it on here, or you can turn it on by checking the box. You don't need to create an overlay to use that tool.
So Tim, is
Great.
this a new tool which has been added? Because I too was not very familiar with this one. You're looking at two parameters and looking at the
Right.
third on the two parameters, right? Yeah, it looks
That is
quite different.
correct. And you can see if you pick the same color, you can see the scale. So perforin on that scale, you can see how it's just using the range, and the higher, more redshifted regions are reflecting greater positivity. And so if we choose a different color, like let's do CD38, oh, well, I already had that, DR, it's showing me where those in the enrichment of a third antigen is within this two-dimensional bivariate plot. And it's underwhelming in a lot of cases when you're looking at raw data, but when you start doing dimensionality reduction and creating dimensionally reduced continents, then turning on the color axis actually shows you exactly where your T cells are, your B cells are, and other subpopulations within that representation of your data.
Okay. Okay. So I think Karthik, you got your answer. So we move next to the next question from Sandeep. Which gate would be ideal for the samples with low expression of markers?
With low expression of markers, well, you can always use a histogram, right, and set a threshold over which you want to call positive. And that might be acceptable, and you can always go to your customize axis and then change that width basis to spread out your negatives a little better to get that where the positive starts and the negative ends. But there's no rule as to what would be a better gate. If you're on a bivariate plot, any one of those gating tools will work to gate a low expressing population.
Mm-hmm. Yes.
That so there isn't any rule that says that there's one gate, but you do need to know where it ends, right? Where the negative ends and where the positive begins. And so let's actually scroll through the samples here real quick. This is an unstained sample, but it has phospho-ERK in the mix. And so if I go to my CD8 population here, actually, let's just go down here. I'm working on the root file. If I go to my CD8, you notice phospho-ERK has a little bit of expression. If I go to the next sample with these little scroll bars, they all become positive because this is a stimulated sample and phospho-ERK has gone up. But if I go back up the list, you will notice that there is going to be a sample here which has no phospho-ERK. So no CD4, no CD38, there's no ERK. So no ERK, no phospho-ERK. This is the sample, the FMO, where I've left out the phospho-ERK antibody completely out of the mix. And this can tell me where my true negative is. So I expect when I draw a gate, even if it's a low level of expression, if I have an FMO that dropped that antibody out, so I'm looking at all the other antibodies, but not this one, I would hope that this gate is nice and clean, and that gives me a understanding of where the true negative is. And then when the antibody is added, then you see the actual positive expression. And so my answer would be any gate is possible, even for low expressed antigens. But when you have something that is dim, or is that a continuous streaky expression pattern, like a secondary or tertiary antigen that is a very low level expression, one thing that you can consider doing is running an FMO control and leaving just that one reagent out. And that way, when you gate down to your population of interest and look at that distribution, see where the true negative is, and then when you add the antibody, you can see it actually appear, the expression pattern appear, right? The difference there. Without an FMO, you're kind of just guessing if it's really low level or if it's a streak, where to place that gate for positivity. It's an arbitrary decision. Hope that makes sense.
Yes, great. Thank you, Tim. So we'll take a couple questions more till the break continues. So
Sure.
next one comes from Karuna, who is asking, what is the use of parameter key option while creating sample groups?
Oh, that's a really good, and it's something I just skipped for the sake of time and moving on. So the keyword band under the workspace tab has this option to create groups from keyword, and keyword column, right, that has some information in it, such as my stimulation conditions here. Create group from, create automatically groups corresponding to each of these individual labels as the inclusion criteria. So I have 4 different labels here, and it will create 4 groups automatically and put the samples in to those groups. So create keyword, oh, not that one, sorry. Here, create group from keyword value, select it, group from keyword value. And what it did was automatically make 4 groups and put all the samples from this group based on the label here into the proper group based on that label. And if I double click on that group, you will notice that it has automatically set the sample inclusion criteria to be equal to a particular value in that column. And
Okay,
so
I think,
what this
I think
means,
if you, okay, sorry,
if you have these labeled automatically and you want to create groups from them, I can just display the keyword column, select a keyword label, and create groups, and then they automatically come out as the inclusion criteria. What are you going to say, Milan? I'm sorry.
yes.
Was I off the mark there for the?
No. I'm sorry. I just went into in between. So if you double click on any group, just to see to modify option.
Yeah.
Then I can probably show you. Yeah, on the top right, there's an option parameter key. Yes, that's that I think that is the question which Karuna is asking.
That is a good question. Nobody's ever asked it for me. I never use it. It's totally true. I mean, you just stumped me. Congratulations. I've never used that function, and I would have to ask somebody else how do you use parameter key, because I don't use it at all.
Okay.
My apologies for not having an answer to that one. That is a new one for me.
Okay. No problem. No problems. Okay. It's okay.
Anything else?
Peter, do we take more questions, or we discuss later? We continue with the session.
I think we have a mentee question, but I think we have no time now. I think we'll have to. Oh, what about Tim? What about your break? Do you need a break, or you
No,
can?
I'm fine. Totally fine.
Okay. Yeah. Thanks.
No,
so let's
coffee.
continue.
I'll never go to sleep tonight.
What time is it, Tim, over there for you now?
It's coming up on 10 p.m.
Okay, you got another 2 hours to go, Tim.
Okay. I'll get there. It's okay.
Yeah, thanks, Tim. You can continue, Tim. Thank you much.
So, thank you guys. Welcome back. And as we've gotten our gating down and we've dealt with group gates and such, so let's go ahead and take a look at the samples. You're going to want to review your gating, right? And you can do that at any level within the graph window by double clicking, as I've shown you to show that graph, and then scrolling through the samples one by one and seeing the, what the effect of your different conditions are, what they look like. And then, as I said, if you want to synchronize the gates by clicking that synchronize button, then you can move the gating anywhere you want, and it'll automatically update to all the samples. So you can make adjustments on single samples. But when you start drawing a whole bunch of gates and you have all of these different levels to look at, doing this review one level at a time within the graph window is quite inefficient. And so that's when I start looking into my layout editor for the review step. So the layout editor is this extra window that will pop open. And within this window, what we will do is drag and drop gates from our gating hierarchy, or you can actually send them to the graph window by right-clicking, or to the layout editor by right-clicking on the graph window and copying to the layout, and it sends the gate there. So either way, either drag and drop your gated population, or right-click within the graph window and send to layout editor. This gives us a visualization, right? And it's always going to show you the last 2 parameters you were working on when I was gating here. So it's going to show me those to start in the layout editor. And I will actually take my CD8 population here. So there's my CD8s for this sample. What I will do here is think about a couple motions here. A right click on a graph plot will give you a list of options of things that you can do with that plot. That could be copy and paste options, or you can go and modify the properties or the formatting of the plot. You can also toggle on certain features like adjunct histograms, which show one-dimensional histograms on the edge of the 2D plot, or you can show your ancestry. That's the simplest way to get at how you got this population, how you gated to display this population. So these are thumbnail images showing the entire gating hierarchy leading to this plot of interest. Or you can show backgating. And backgating is a special type of gating where we take the population that we're looking at here, CD8 cells, and backgating at each level is showing the red dots are where your CD8 cells in your final gate would be if you had removed one gate at a time. And if you zoom in here, I'll show you what that means. So singlets, lymphocytes live minus CD3 positive and then gated on the CD8s. What this is telling me is that if I removed this CD3 positive gate and left all my other gating, but just took away CD3 positive, went right from live to quadrant gating, I would end up with all of these additional red events in my final CD8 gate that I'm displaying. So this is showing me any red events that are outside this gate. That's what was cleaned up with my gating compared to all of the other gates without that single one. What this does is give you a mechanism to see where enrichment is, if there's a population that you're cutting through on the border and you're missing some events, to really see how good your gating was, and it can help you interpret the quality of your gating. So those are right-click options. You can also make multi-graph overlays, histograms, multi-graph color mapping, all parameters by another parameter, or an n by n half matrix plot. So that's right-clicking to show property options. Then you can open up your properties and do additional formatting here by right-clicking and selecting the properties option, or a quick double-click on the plot will open this color palette. So I can get to it in both mechanisms, properties. This palette has what we call the graph definition window, and it has 4 different tabs, and you can modify the features of this plot. You can change the type of graph here. Maybe I do want to look at it as a contour plot, showing a grid in the background and using large dots. Click apply will apply those, right? Showing the outliers will give me some additional outliers. Large dots overemphasizes the outliers, which is nice if you're looking at very rare populations and you want to show those outliers more distinctly. You can change the color here and the parameters. The annotate tab allows you to show or hide various features, so I can show or hide all the axis information or different pieces of the axes, like hide the numbers or hide the label. You can also override the label here by typing in a different label. So maybe I want to call this just PERC or phospho-ERC. Instead of showing the entire comp Alexa 488 phospho-ERC label, it will just override that and show phospho-ERC. Maybe those labels are too small. Well, you can go to your fonts tab and change the axis labels from 11 point to 18 point. Click apply, right, making the axis labels bigger. And then the legend is you can show or hide a legend that will be automatically shown with an overlay, but you can add keywords and other statistics or even drag statistics in to create legend columns here. And so these are all of the annotation features that are available, or the graph properties. And again, you can get to that by right-clicking, go into properties, or just a quick double-click on your plot will open up that set of options. Within this layout editor, it has its own set of tabs up here. So when you first open the layout editor, you'll get a blank tab. You can double-click on the name and give it to a basic name, CD8 T cells, as descriptive to tell you what is going to be in that layout. And then you can create as many layouts as you want clicking this plus button and renaming them to appropriate names, and then dragging in one or more plots. You don't have to do them one at a time, but you can, and positioning them and organizing them the way that you want. And so what I might do out here, I've got 3 different markers within my CD8 positive populations. I'll show the ancestry instead of the backgating. I can make this horizontal with a right click and move it up here, make it bigger, double click here and change this contour back to pseudo color because I would prefer that. And then maybe I'll duplicate this plot since I made some formatting changes and added some stuff. I'm not just going to drag it over again. I just want to show a different set of parameters here. So I can go ahead and right click and duplicate this plot, move it over. Maybe I'll delete that extra set of thumbnails. Zoom buttons are in the bottom left-hand corner, and page breaks can be grabbed and moved. They can also be hidden under the Edit tab, show or hide page breaks if you're never going to use them. But once I've made it, go ahead the file and always to get your drop-down menus here, or you can double-click and go to your specify and change the axes here. In this case, the easiest way, once I've duplicated that, is just to right-click on phospho-ERK and change this to interferon. Right, so now I'm seeing my interferon gamma expression within the CD8s instead of the phospho-ERK. I can do the same thing. I'm just option clicking. You can right-click and copy and paste this and change to one more marker. This is going to be my perforin marker. So right now I've got 3 different markers with all the plots. I've got my history, and I guess I just want to organize things. So I'll select multiples. There's an arrange tab to organize and arrange, space out all these plots appropriately. And then, of course, if I want to make an image out of this layout that I've just created, I can export the image under the File tab as different file types, PNG, JPEG, SVG for scalable vector graphics. Or I can actually batch this image. And before I do that, I'll just add one more feature here. These are drawing tools in the middle section, and I want to add a text box here. So I'm just going to click the A button, add a text box. And if I type in information, this is my text. It's really small here, but that's just static information within that text box. If you right click on a text box, you can insert keyword information. And so I'm working with this patient sample LD2, insert keyword. Oh, there we go. There we go. Okay. Insert keyword for my sample LT2. PI is the one that I'm looking at right now. And this is my keyword list, right? These are all this metadata features. Well, what's the easiest thing for me to see here? I want to look at the patient ID and the stim condition, so I can insert both of those as actual labels within the text box, make them big, go into my text options here and making them large. I can always use my object tab to remove the line or fill or anything that I don't want, so I'll remove no line. And what this is doing is making a label that says what patient and what stim condition I'm looking at and associating these visuals with those conditions. Now, the beauty of this is when I actually print this out, it'll be up there as a label, and I can show my boss and they can be like, oh yeah, I can see what I'm working at. Well, I can see what you're working on, what you're working with. Now, the power of the review process in the layout editor comes once you set up this visual representation of your data in a way that is meaningful to you and how you want to present it to, or to actually, if you just want to review all the gating. You can turn on the iteration option and create batch reports within the layout editor. And iteration means we go through a group of samples, one at a time or in a group of samples in an iterative fashion, and display all of these plots that we formatted and the statistics that are displayed there one at a time in a batch process. And so to do this, I'm going to show you first, we want to set the group that we want to look at that contains the samples. I'm just going to focus on the all stain samples that contains my 20 real experimental samples here. And then you need to choose an iterate by option. So it's set to off, which just means by default, which means I can bring in any gating level from any sample and make comparisons here and visuals and just save that as an image. But if I want to iterate through the samples, then I will select this off button and change it to sample. And sample iteration now converts the visual into the first sample that we're working with in the group, right? Because we're looking at the all stain group. It shows what I'm looking at in this drop-down menu. You can select different samples from the menu. It'll display all of the samples in your group, or you can scroll through the samples. So notice I start with the no stim, no ERK, no interferon expression, or low ERK and no interferon expression. Stimulate for 20 minutes to the next sample, and phospho-ERK goes up. Stimulate for 2 hours, and you get interferon gamma as well. Right? And so what I'm doing here as I review these gates is looking for gates that may not be appropriate. And when you find something that you want to change or move or update on a sample, you can right-click on that sample. And again, through this right-click menu, there's an option to open original graph, and that will take you to this level in the gating hierarchy showing these two parameters, and you can make an adjustment. Maybe that was a little too tight for my taste. So I just moved it to the right. Again, because we are synchronized, we don't have to go through and reapply that to other samples. I'm just making a change. And you can go through each sample like this, one at a time, to make changes if necessary. You're like, oh, let me go ahead and open that graph, make a change to the location of that gate. If I want to make a change to a gate that's upstream, I'll press this up button, get to the level that I want to, and make a modification of that gate, right? And so this review step is essential for accurately gating across multiple samples, especially when you're treating them, the animals or the samples of different conditions, and you expect different distributions from sample to sample. If you want to use a master gating approach, you need to apply the gates once and then go ahead and review them. Once I'm happy with that gating, maybe I want to create a report to show to my colleagues. That can be done through this create a batch report button, and that will go through the group by this iteration and show me either a new layout or a go direct to my printer. I can save it as a PowerPoint image or a PDF. And if you go to a new layout, you can iterate across in different columns or rows. And so what this is saying is I want to go through my group of 20 samples and to go down or across and create 3 columns. So how about I've got 5 patients, I'm going to go down and create 5 columns. So what this will end up being is 5 groups of patients, 5 columns that are going down. So sample 1, 2, 3, 4. I'll click the Create Batch Report button, and notice what it's doing. It's creating that batch report. And here's sample 1, 2, 3, and 4. They're all the same patient, LD1, the first 4 samples. Next 4 samples, no stim, 20-minute stim, 2 hours stim, and 2 hours and 20 minutes. So I can zoom out here and you can see this type of iterative process, which gives me this grid of plots. All the plots are the same in their layout, in the way that they were formatted, but each set of plots represents a different sample. And this is another way to go ahead and look through the samples in an efficient way. And at this point, I can now export that image if I would like and save it as a PDF to share with my boss or anyone else. All of these groups of samples are initially grouped together when a batch, but you can always ungroup and get at the individual images if you want to pull one out, or if you would like to open that original graph and make a modification. And so that's one way to use the table editor. This is a gating layout, right, where I show a terminal population, right-click, show an ancestry, and then you can scroll through by sample by using your iterate options to do sample iterations. If we want to make more complex layouts, well, you make a new layout by clicking the plus button. And you can overlay, for example, different populations. I'll just take my CD8s here and drop them on. I'll take my next CD8 population, drop it onto the same plot, and what happens is you get a dot plot overlay. It automatically converts that into an overlay with a legend here showing the different colors. My third sample will be a little more interesting because it's actually stemmed long enough to show some interferon gamma. And my 5th sample is going to be my longest stem, which is very similar, right? So that's a dot plot overlay. Maybe we want to convert that to a histogram. Well, double click on the plot, brings up your specify dot plot. That's not what we want. Let's look at histogram. Apply. Now I've got overlaid histograms. If you want to make the axes normalized, well, I go from auto to modal axis for the y-axis option. Initially, you'll be showing counts. Click apply, and that shows normalizes the peak height. So you aren't looking at different numbers of events here. You're just normalizing to mode. Within this plot, you can right-click, and you can change different types of styles. So I can change the coloring, line weight, or the type of overlay. And I really like to offset these overlays, right? So they're stacked one on top of each other. Or another really nice one, there's a half offset that they kind of layer on top of each other if you want to make comparisons there. And a stagger offset, which is a 3-dimensional view, right? So all these formatting, the line weights can be changed, the colors can be changed through the palette here by clicking on the color icon in the legend of each layer, and the layering can be adjusted once I've created this by grabbing a layer and moving it through the overlay, right? So I can reverse the layering here. You know, so I do a similar thing where I create one view and then duplicate this, change the parameter that I want to look at for the next one, right? So there's a different representation of the same data, and then a third parameter here. Maybe make my keyword labels, do arrangements here, and then you can iterate over this. And in this case, I don't need to iterate by sample because I actually am looking at one patient at a time here for all different conditions, right? So I'm just looking at patient by patient. With this type of overlay, I set this up actually using my LD1 nomenclature, but I'll turn on my panel or keyword iterator, and I just want to hammer this home before moving on. So panel iteration is pretty simple. Sample is one sample at a time. Notice what happens. It just shows me the same sample in all layers, right? They all reference the same first sample. I can still scroll through, but every layer is going to reference that same sample. That's not what I want. So sample iteration doesn't work for this overlay of multiple samples, but panel iteration, this is like saying I want to look at this many tubes at a time in the order of the list here. And so I've got 4 tubes per patient, 4 different samples that have been set up in this overlay. So right now it doesn't work. I'm only showing 1 sample at a time, but maybe I want to see 2 or 3 or 4. So now it's going to go through and show me my first 4 tubes and then the next 4 tubes. Notice I'm showing my list here is now every 5th tube because I'm showing 4 at a time. And so my iteration over panel is looking at this many tubes at a time through this list, and you can scroll through those samples and you can see the data update and the keyword label of the patient ID update to know what I'm looking at as I go through all of this. The last option for making this type of iteration work when you have multiple samples and you're comparing them is to not do panel iteration, which depends on the order of the workspace list, but do keyword iteration. And again, this is where I go back to my keyword and say, once you start using them, you'll never go back to not having any labels. Because this keyword iterator, it prompts you to select 2 keywords. The first keyword has to be something that's the same across all the tubes you're displaying on the page. And that, in this case, is the patient ID, right? All the tubes have the same patient identifier. The second discriminating key has to be something that's different across these 4 tubes displayed on the page, but is repeated over the iterator. And so my stim condition keyword is there's 4 of them per patient, right? 4 different tubes of stim per patient. And so now when I iterate by stim and discriminate by my patient IDs, remember that these are stim condition and patient IDs here, then I get a drop-down list of just the patient IDs, and you can scroll through them, and everything is based on the keyword labels. And so if I reorganize this sort order here, it's not going to affect my layout because I'm pulling the information based on this combination of metadata labels. The patient ID plus the stim condition gives me a particular reference to a sample. So keyword iteration can be very powerful. I always like to push this out to a web page. If you're doing an external batch report, the destination is the location of where you'll save. And I can click that batch report. In this case, with a web page report, it makes PNG images and an HTML link, so it pulls them automatically up into my browser. And I can scroll through them, and those images have already been exported as PNGs. So this is going to be my histogram layout. Histogram overlays, right? So just as a summary, the layout editor is accessed from this L button. You can make as many layouts as you want by clicking the plus button here. To get data into the layout editor, you select populations and drag and drop them into the layout. To modify the visual representation of that data, you can right-click and select additional options down here, or go to your Properties tab and change the type of graph and the formatting within these different tabs, which control the visualization here. Once you've set your properties, you can then turn on your iterate by option. The standard one for reviewing gating is to go by sample and look through each sample for your different distributions and make adjustments by right-clicking, opening the original graph, making a modification here to the gating that you want to adjust. Once you're happy with your visualization, You can create a batch report to an external web page, or you can create a PowerPoint or a PDF or a web page, or you can just create a new report in your layout that is iterating across this group that you've selected by this iteration and showing you this set of plots in a new batch layout, which then again you can go to File and export as an image. Now, all of these options can be used in combination with each other, but those are the core that you're going to use in almost every single analysis, is reviewing gating initially and then making some focused descriptions or representations of your data to go along with the story that you're presenting to your audience in a paper or presentation. So once I'm done with that review, I'm happy with my gates. The last thing is to push out all my numbers. I haven't added statistics yet, so I'll just add those really quick by clicking this Add Statistics button. I have the CD8 population. I just want to show it. I'm going to add phospho, ERK, and perforin MFIs. Click Add. Right? They become gray nodes here that can be, just like gating, can be applied to a group by dragging and dropping or copying to a group with the right-click mechanism. Now they're red. They're on every single sample, and I can enumerate those just like a gate. If you do drag a statistic in, it'll actually create a text box linking that statistic into a text box with the information in it. So you could, for example, place a label with a statistic on another plot if you wanted to enumerate that. There are other ways you can do it, but obviously I'm going longer than I should and just running out of time. So let's wrap that up and say the layout editor is a great way to display multiple plots and to create images for publication and presentation by exporting the displayed image in the proper format here. Once I'm happy with all my gating, my last step is really to get these numbers out of FlowJo in some usable format. The easiest way to do that is not to transcribe them or copy them down, but to use your table editor. And the table editor is similar to the layout editor in that it opens up a new window. It's accessed from this latticework looking icon. And what we do is similar to drag and drop populations or statistics that we want to enumerate from our gating hierarchy into the table editor. Each time I do that, I'll take my live, CD3, all my individuals, my median of ERK, and yeah, we'll just stick with that. So I'm selecting multiples, but just keeping it limited. Drop these over here, and each line item is a different gated population. It'll show you the statistic that you're working with, and then the parameter, if necessary, describing for what that statistic represents. The table iteration is almost always going to be by sample unless you go and do some fancy options like adding a column and creating a formula like a ratio between samples. So that's automatically set to sample. And the easiest thing to do is just click the Create Table tab once you bring in your statistics that you want to see in this table. And like the layout editor, it goes through the group of samples that you specify here, and you can choose different groups to focus your report on a subset of samples by sample and show this set of statistics in a displayed table, or you can choose to send this to a layout, the image, or to a file of your choosing, like a text file, a CSV, or an SQL database, whichever you need in this location. So I'm just going to show the display. That's usually what I do, create the table to display. And what you're going to notice here is that each row in the table editor becomes a column in the output. And then the rows in the output table are the statistics represented by this gating tree and the statistic across all of the samples in the order of the sample list. So here's my LD1, 2, 3, 14, same order, LD1, 2, 3, 14, such. And each one of these columns represents the frequency or the MFI depending on the type of statistic or gate that I drag in. Now you might notice that these gates have really long, these columns have really long header names, including the entire gating tree. So when you start doing really deep gating trees, especially on high parameter data, these get kind of unwieldy to use, just very long string names. So do know that you can add your own names here by double clicking into the name field. I can just call these live or call the next one CD3. Right, give it a unique name, and then when you create the table, it'll actually use that name instead of the entire gating sequence. And we've actually added a kind of auto create names option here under your edit tab. that you can choose to add. I'll add the last population name and the statistic and click OK, and that creates this string of information that becomes the column header names instead of the entire long gating structure and statistics. So a lot simpler to work with these named fields downstream. The other thing I like to do is each of these columns, there's variation here, but the just black and white formatting doesn't show my brain that there's differences. You have to read the numbers to see those. Do know that you can actually add visualization under this Visualize tab to any set of columns, one at a time or in combination. I like to use the heat map option, and when I apply that visualization, it'll show an icon of what's going to be applied. Creating the table now individually heat maps each column with its own yellow to blue, high to low heat mapping, and immediately all the features and the changes start jumping out to me, right? I can see that my frequency of live events was pretty low for LD2. Knows I need to go and check on the fight calling ability of this of whoever processed this patient sample. I can see the repetitive nature of the stimulations where phospho-ERK goes up right away and stays high in all of the stimmed samples. Interferon takes longer to go up but is very high and shows variation between patients in the longer stims. And then perforin is relatively stable across the stimulated conditions but varies between patients, right? So visual formatting can really do a lot for your brain to understand what was going on in your sample. You can always copy this by just Command A, copy it out to Excel, and bring the data over that way. So I'm just copying the table from the display and pasting it, right? There's my table information. The other option is to actually save this table to a file. And I don't want to leave out the fact that you can actually add keywords here under the Edit tab. I can add my descriptive keywords. So if I want to make comparisons across my stim conditions, I will add that in. Maybe I also need to compare my patient IDs, so I'll add the. And when I now get out the table that I'm creating, all of those values associated with the samples will also be shown here, and it'll make it really easy for them to make comparison graphs and summary charts to look at these different conditions for any given marker downstream using Excel or Prism. So that's the final step there, is pushing out all of our numbers. Maybe you just want to go to File and make a CSV, a text file, and save it somewhere. That's fine too, but oftentimes I like to look at the data first, so I add the visualization first and then display the table to see what's going on. And if I want to put that in that table with the formatting, the color formatting, I can go ahead and change that display to put it to my current layout and then create the table, and it shows this table now within my layout. And I could add that as a visual feature with all the numbers and make a image out of it, right? Just share with somebody. Okay, so I'm sorry, I never went back to my presentation. I'll run through it here in just a moment. But last two things. Now, I've already saved this. File, save as a workspace. The workspace save saves a workspace, which is right here. I just didn't left the name as the date. So now I can just Command S, and it will automatically save to the same location. If you save a workspace, it doesn't automatically create a duplicate of your data. This workspace is still referencing the files contained within my, here it is, my PFIX data folder. If you want to zip all the data along with the FCS files into a portable unit, I would recommend using the archived ACS save. That creates a zip file that includes a copy of your original data files, and so you can just move the ACS around and put it on a thumb drive, pass it to a colleague, and you will never disconnect the data, the raw original data, from your workspace itself. The other save option that I'll promote is this export as a template. So I'll just go ahead and do it. Export as a template. It creates a WSPT file, not a WSP. I'll just put it on my desktop here. So there's my template. And I've already saved this workspace, so I'll go ahead and close it. And I'm going to now open my template. So I'm opening my template file. Now the template saves all of the structure and organization that you've created with your gating, your layouts, especially if they're batched and they're just waiting for data to come in. and your tables, right? So you already have all the structure there. You're just not referencing any data. So if you run the same experiment week to week, you can set all this analysis up on your first experiment and then come in with your FCS files, drop them into this workspace. Because I've used these inclusion criteria with live groups, it automatically picks up the right samples into the different groups. The gating gets applied to any samples that go into my gating group. The statistics enumerate, and then I'm already off to the races with creating a table for my analysis or iterating and creating a batch report within the table editor for my various purposes, right? So this is a great way, file, save as a template for reproducing the same analysis from week to week on different samples if you're doing batches of runs and analysis. So I know that was a lot of information, and I hope that you will get a chance in this week to kind of review and click around and test this out. I know you're all getting access to FlowJo, and I encourage you to take some of your own data, or you can go to our website and get the basic tutorial data, which is 8 of the samples I'm showing you right here are in that tutorial or given out, and you can load them up and work with this exact same data. So all of the slides that I skipped are showing you the exact same things: how to work in the layout editor, how you right click and show various options, double click to change the parameters or the formatting and properties of your graph plots. And then importantly, the batch analysis of layout editor graphics allows you to iterate across a set of samples to create a batch report showing you all of your various features that you're interested in. And then you can save that or export that as an image of various image types. The table editor is very similar, right? Opens up a new window. You drag and drop all your sample gates or statistics that you've enumerated on a single sample into the window, and then use the iteration and batch options that are available there to create an actual table report that you can take outside of FlowJo or use as a visual in the end. Templates, simply just saving all the analysis without actually referencing the original files so that you can drop new files in. And an ACS zip file is a creation of a package that includes a copy of your original data that is then referenced by the workspace. A normal workspace save only saves the XML and then brings in the original samples. And the only problem with that is that if you, if your samples get moved to a different location or get disconnected from the workspace, they might be missing, and you can always reconnect them. If you have a question about that, don't hesitate to reach out to me or to my tech support team. Preferences, I didn't go through a whole bunch of them today for time constraints, but there are all these different areas of the program. The most important to me are font sizes and colors, so gates and fonts preferences, but there are a number of preferences, and we'll go into another one, the diagnostics pane, to set up our plugins here in another half an hour or so. But if you have questions, don't forget that we have the searchable documentation available, and you can get to that just by clicking on this little question mark icon in the top right hand corner of the workspace, and then you can search for any function and it'll give you pages to understand that function better. We have a FlowJo University group of area of our website that has presentation decks and videos that you can watch on various topics, and then our technical support team, a great set of guys working hard to solve problems and find solutions for you. If you ever have a question and I'm not available personally, don't hesitate to reach out to flowjo@bd.com to get an answer there, and we'll get back to you as soon as possible. So with that, I'm going to pivot away from this topic into a different topic, compensation. Does anyone have a question or a comment from the team here leading this at the moment?
Tim, I think we can take some time off and go for a live poll. If we have some questions for the participants they can go with the live poll and answer some of the questions, and then you can probably continue with the compensation. Right, Peter?
Sounds good.
That's okay, Amar. I think we can go ahead with the live polls.
The question one is live, sir, now. I hope you can see it on your screen. Yeah, it's in the chat window, so everybody can access it and push in your answers.
Ankita, could you read out the question?
Sure, sir. The question was transformation in FlowJo. The options we had given were accepts visual display and scaling of data. Second, does not affect raw values or statistics. Third, gets rid of squishing of cells. And fourth was all of the above. We have started seeing responses. We have 15 responses so far, and all of them are saying that, and 17, 18, all of above. So Amlan, would you like to speak further? Yeah.
Good job. And that is the correct answer, right?
Yeah, good
Transformation is all about rescaling, but it is a underlying.
Good job, team.
Thank you all. So we have how many more questions? 3 for this break.
Yes, the second question is live now, Amlan.
Okay.
Yeah, so let me read out the second question. Is this the one? Table editor and layout editor in FlowJo?
Yeah, so there's been a typo. I think we have shared the correct answer, so we can move with the next. If we have any discussion we want to do over it, we can do, but the answer is already written there, so that's a typo from us.
But I think, I mean, we can just quickly discuss this. Tim,
Sure,
would you like to go?
What was the question? I'm not looking at the, because I'm displaying my screen.
So
I'll
do it for you. So the question is table editor and layout editor in FlowJo are. Basically, the options given were first was so you can see the answer is both B and C. So the it will be table editor is a tool for creating statistical reports, and layout editor is a tool for graphical reports. Okay, and C is table editor allows to add visual formatting like we saw in the heat map. Right now on the screen also background you can see, and create batch report. Layout editor has a functionality where you can create batch reports. So the correct answer would be option D, which is both B and C are correct.
Excellent.
Okay.
Okay, we can go to the next question, I guess.
Ankita, next question.
Yes, sir, it's live now. The next question, Amlan.
Okay. So next question is, what does a live group checkbox under the creating and editing group options in FlowJo is meant for? So the options are, it is for highlighting samples of a particular group currently being used for analysis by researcher. Second option is automatically including samples based on the user-defined sample inclusion criteria. Third option is automatically selecting live events and removing dead events or cell as part of the data cleanup. And last option is both B and C.
Drum roll.
Ankita, are we getting responses?
Yes, we have 16 responses, 17 for both B and C.
Ah, why do they think it will automatically clean up the data?
Nancy has mentioned B. Okay, we are seeing multiple. So 25% say A, 17.5 we have for B, and mostly we have for D, that is both B and C.
Okay, I will request don't rush, just read thoroughly the answers and then decide.
Amlan is
Nobody's
giving us
getting
a hint.
graded on this anyway, so it's not a problem. But you know, it does not clean up samples. It does use the sample inclusion criteria, making this group live. If you add more samples, it will look through those samples if the group is live and automatically add them if they fit the criteria.
Now most of them are going with A, Amlan. I think they listen to you and it's Yeah, so
But they
there's
can see the option at the background. Tim is showing up his screen and it's
Yeah, I am. I'm
definitely
like pointing to it.
pointing to the right answer.
Cheater, cheater.
oh, Tim is helping us
You
all.
wish the instructor gave you all the answers to the test.
And the last question, Ankita, now?
I think this is the fourth one, right?
This was the last one, Ankita?
No, we have one
There's
more.
one
The
more. We
fourth
have one
one.
more. This was the third one, yeah.
Okay.
Yeah, it's live now. The fourth and the last question for this break is live now.
Oh. So I will read out the question for all of you. Which among below saving option in FlowJo saves both the analysis and data files together in a zipped file? Option A is workspace, WSP. Option B is archived cytometry standard or ACS. And option C is workspace template, WSPT.
I think we have just done it. Tim has just done this.
Yes. Yes.
Yeah.
So all of us are giving the answer as B.
Okay,
Yes.
that's correct.
Okay. So I think, thank you, Ankita. I think we can continue.
All right.
Yes.
Yeah.
Thank you guys so much for putting those together. And I think it's always good to do a sanity and knowledge check. Again, if you have questions, don't hesitate to type them in, and we can certainly prepare a list. I can prepare a list of answers and go over those in our next session if we don't have time to finish up everything
Yes,
today, okay?
I think we have gathered a lot of questions, and the answers to all of which is not for time crunch will not be able to give now, but we will get back to you with maybe a week's time or 2 weeks time. We'll compile everything, all the answers, and we'll get back to you.
Yeah, I think it's really important to make sure that they get all that
Yes.
information and
And
clarification.
Tim, for you, I mean, it's continuously for 2 hours in a row now, and you have still 1 more hour to go. You want to take a break, then please
I'm
go
fine.
ahead.
No, I prepped.
Okay.
I'm good.
Okay.
I want to get as much time in as I can for demonstration purposes. I know it takes a lot longer when I actually point and click and show you everything that I'm doing, but I think it's important to show the actual interaction, and a lot of these functions makes a lot more sense than just a slide. So thank you guys for your time. So I'm going to move quickly into compensation, and my goal initially here is to just show you how compensation works, but I'm also going to mention some new compensation tools that are available, some new methodologies that we've employed just in the past year, which might make life a little bit easier, especially when you have lots of controls when you go into high parameter data space. So the outline of this section is I want you to understand what compensation is doing, and then introduce you to the traditional way of doing compensation, illustrate that for you in the workspace, and then also mention this AutoSpill compensation algorithm, which has recently been integrated into FlowJo. And so let's talk first, what is compensation doing, right? It's correcting for fluorescence spillover between channels. When you acquire data without compensation, you will see interaction between fluorochromes. In this case, I'm showing you an Alexa 700 stained cell compensation control. That's one color stained sample, cells. And you can see the skewing of the population into this APCH7 detector. This is because the Alexa 700 emission spectra actually overlaps the detector for the APCH7 channel. And so you get some photons from this fluorochrome bleeding into the detector for another secondary channel. What we need to do is correct for this. And in FlowJo, you can know immediately if your compensation, if your samples have compensation, and if you're looking at compensated data, because you will have a comp prefix in front of the parameter name if you're looking at compensated channels. Without the comp prefix, you're looking at the raw data, and we can toggle back and forth between those if we would like to see original raw versus compensated or corrected data. When you compensate, you really have to have a single stain control for each fluorescent probe. That's an antibody fluorochrome conjugate or an intercalating dye or whatever you're using. And you're isolating the emission spectra of that fluorescent reagent and determining how it spills over into all the other detectors. we create a matrix of these values and then apply that matrix to each cell individually to correct for these overlapping spectra. And what you will find when we apply compensation is that the positive population that was skewing into the secondary detector here will be brought down, and the positive and negative populations will have equal medians in that spillover channel if it's a correctly compensated sample. And so you can use this principle to actually double check that your compensation is correct by compensating your individual single color controls and then taking the median statistic of the positive and negative and trying to match those if you need to edit any matrices. So again, compensation corrects for spillover. It's applied on a per event basis, not on a population or sample level basis. Each independent event based on its combination of expression is corrected for some amount. Spillover, I said this term a couple times in that first slide. Spillover is the emission of a fluorochrome. This is showing the emission spectra of PeSci 5.5, and we're picking up and counting the photons in a primary detector that is situated around the peak emission of this fluorochrome. There are also on our instruments a number of extra other detectors that are going to pick up other reagents as their peak, and we call these secondary detectors. The primary is the one centered on the peak, and all the other ones are secondary. And you can see the emission spectra of PeSci 5 actually bleeds into these different secondary detectors by some amount, and that's what we're going to try and do, is remove this spillover of the fluorochrome into the contribution of this fluorochrome into all its secondary detectors. What we do is take a single color control and we measure the fluorescence and we determine how much signal is spilling into each of these detectors, and then we create a matrix to compensate out or remove the spilled over signal. Compensation gets more complex, of course, as you add additional fluorochromes. You can see now my Percy PeSci 5.5 is bleeding into PeSci 7. PeSci 7 has bleed in the PE channel. FITSI is bleeding into PE. PE is bleeding into FITSI, right? We have all these different interactions. And so, again, we need to isolate each emission spectrum initially to determine how that spectrum bleeds into all the detectors on the instrument, and then we create a matrix and solve a series of linear equations to get the compensation matrix. To simplify that visually, what is compensation? What we're measuring is the fluorescence across, we're measuring the fluorescence spectrum across all these different detectors. And the primary detector for each color, and these are different detectors, and these are different fluorescent reagents. I'm simplifying this down to a 3-color experiment now. FL1 emits in the D1 most, and then it has about 12%. It's scaled based on the maximum emission here. About 12% of the photons that it sees in D1 are actually spilling over into D2 and 1% into D3. Here, FL2, right, it maximum primary detector is D2, it spills 15% and 20% into D1 and D3, and then FL3, which is the third reagent, spilling 1% into detector 3, nothing into detector 2. And so what we measure here is actually the spillover into each of these detectors, but we only know the Ds. We don't know the entire combination here. And so what I want to do is pivot this visualization 90 degrees. So now we're looking at the emission in columns and the detectors in rows. So when we do an experiment, we only know the Ds, right? We measure D1, D2, D3, and it creates this series of linear equations that one is the entire 100%, 0.15 is the amount that spills that's contributed from FL2 into detector 1, and 0.001 is the amount of signal that we detect in D2 that's coming from FL3, and we need to actually solve for these different FLs. So to do this, it actually requires this matrix math where we convert into a matrix, a spillover matrix, that we do a series of linear equation to solve for the inverted matrix. And I know this is a lot of math, and I don't expect anybody to understand it, but if you've ever actually done matrix math, beyond a 3x3 matrix, you never want to invert a matrix by hand. It'll take you way too long. Let the computer do it. So we have just 3 fluorochromes and 3 detectors here. Imagine now we get into 20, 25 colors. This matrix becomes absolutely huge, and the math is not solvable by human manipulation. You have to plug it into a calculation. So what we measure, bottom line, is the spillover, and we take that spillover coefficient matrix and do an inversion to get the compensation matrix, and that's what we correct for on a per event basis using this entire matrix of spillover coefficients. Now, when we do these measurements, we have to have a single color control for each fluorescent reagent in the experiment. That is the #1 rule of compensation, is to have single color controls. FL1, FL2, FL3, so we can see how each of these fluorochrome emission spectra bleeds into each detector. Additionally, if you want really good compensation, you should follow these 4 rules of good compensation controls that have been around for about 10 years now. Compensation controls have to match the exact experimental fluorochrome. That means you can't compensate a GFP mouse with a FITC antibody. That's not appropriate. It's a slightly different fluorescent signature. So you should always use the same fluorochrome. Background fluorescence should be the same for the positive and negative control. For each single color control, we're actually going to gate positive and negative events, and you have to have the same particle type to make the calculation correctly. If you mix and match beads and cells as the positive and negative, they have different autofluorescence levels, and so the measurement that we're going to make is going to be off. And so always, if you're going to stain with beads as the positive, use beads as the negative. You stain cells as the positive, use the same type of cells as the negative, so the background autofluorescence matches. The control should be at least as bright or brighter than any sample the compensation will be applied to. If you have really dim comp controls and very bright samples, you can get poor compensation above that level, and so one of the rules is to make your compensation nice and bright. Again, beads are a great solution for this because you can make the beads as bright as you need to by adding a little more antibody when you stain them. When you're using cell comps, you never know which one is going to be brightest, especially with patient samples that come in from a clinic or something, and you never know who's going to have that really bright phenotype, and you would not know which one to use for the compensation control. So beads are easier. And if you're using fixed perm, this comes in when you're using harsh buffers to get it nuclear, to get down into the nucleus for phosphoflow, or if you go in or looking at cytokines, you need to fix and permeabilize the cell membranes to get the antibodies in to detect the antigen. And so sometimes, depending on the fluorochromes you're using, fixatives and permeabilization buffers can alter or break down the fluorochrome. And so if you're going to fix perm your cells, you might as well fix and perm your compensation controls identically to the cells so that everything normalizes and you don't get any breakdown. Again, it depends on the buffer system here, but this is, if you do start seeing some weird compensation issues with your intracellular cytokine staining, this could be an issue. So if you follow those rules, you'll get nice, bright, positive controls, and you can bring them into FlowJo, and they'll automatically load into the compensation group. The compensation group itself right here in red, right, has 12 compensation controls, and they're shown here as bead comps or cell comps. These are the same samples I was using before. And when you load your samples, you may have already calculated compensation on the instrument, and you don't necessarily need to recalculate compensation within FlowJo if you trust the compensation that you've already created. You'll know if there's compensation applied just by looking at this little square badge icon next to the sample names. You'll notice that the square badge of the compensation controls is empty. There's no lattice work or coloring to it. And that means that there's no compensation applied to these samples. If you see a gray lattice work filled in like a little grid, then you know that compensation is already present on those files. And the easiest way to view your compensation is just double-clicking on the square badge. Any of these square badges will bring you to this matrix preview window. And if you have acquisition-defined compensation from the instrument, it'll show up as a gray matrix that's already present, and you'll see the values of your compensation matrix. The way we read a matrix is that this fluorochrome, let's take Alexis 700 again, is bleeding into this detector, APCH7, by some amount, right? And that is the correction that we're going to make for every given units of fluorescence in the Alexis 700 channel. We're going to remove this many units from the APCH7 channel. And so you can always say the rose fluorochrome is bleeding into the columns detector by some amount. Down below this matrix preview is a preview sample option where you can look at a half matrix, an n by n set of plots, and you can choose to preview any sample by selecting from the drop-down list and any population that's gated on that sample to view the effect of compensation. This compensation, when it's applied, the events look like they're gray. So gray is the compensated data, blue is the uncompensated data, and you can see the corrected or shifted data from blue to gray with this overlay here, which can visually tell you if they're skewing one way or another. You may have had poor compensation. But this is called the matrix preview window. We also use this window to apply compensation. If I want to apply any of the matrices that I've created here, the traditional compensation, AutoSpill compensation, or even spectral compensation, I can select that item with a different matrix color and grab this M button, dragging it to a sample or to a group of samples, and that matrix will be applied, and the color of the badge will change to show me that I've applied a green matrix or a pink matrix or a blue matrix as appropriate. So these could be color coded. If you have multiple runs or multiple panels within the same workspace, you can color code the matrices and choose different colors for them and immediately get a visual cue of what compensation is applied to what. So easiest thing to do is just double click, see what your matrix looks like, and then you can modify it if necessary or create a new one, which is what I'm going to go through here. So initiating compensation, the first thing you need is your compensation controls, all your single color stain controls to go into your group. Then once you highlight that compensation control group, it'll open up the compensation platform tool, and you'll see this little overlaid emission spectra looking. This is showing the spillover between two emission spectra. And you click that button and it'll start by auto-gating your samples for size, that is gating on the major population to get rid of all of the additional other subpopulations in here. You don't want to gate on debris, you want to gate on cells, and you want to gate on a single population. And so it'll auto-gate usually on your lymphocytes if it's a cell comp, or on the major population of beads if you've stained beads for bead comps. And then it'll auto-gate on the positive fraction within that subset, right? So size gated, and then the auto-gating for the positive fraction within the size gated events here. And so you'll see these gates appear downstream of each of your compensation controls automatically. FlowJo is looking for the brightest sample in that color and calling it the positive control, and then it's auto-gating it. This window will pop up and FlowJo will fill in these samples as with the names of the files that it believes is the brightest channel. It's looking to match the channel name within the sample name. So if you have that match, it gives you a nice green badge, says, oh, look, I found the sample. If there's not the perfect spelling, your parameter is a little bit different. This says AX488 versus A488 is the parameter name that was typed here. It'll give you a yellow warning saying, oh, I don't see the name, but that's fine. You can still move forward. My job here is now to review the auto gating for positive and negative and size, and make sure that it's appropriate. Immediately, I have an issue here where the ARD sample didn't auto-gate on a positive fraction because the size gating was on the good cells, and probably most of the dead cells are a little lower on forward scatter. So in order to make an adjustment to this gating, I can double-click on any of these preview windows. It'll open a graph window, and I can make an adjustment to the gate. You can pick and choose which parameters you use for compensation from the drop-down menus here, or you can remove parameters if you don't want to consider them. For example, I have a blank channel here, so I would remove that before calculating the matrix and finalizing that matrix calculation. You can use internal negatives. That means the same single tube with both a positive and a negative, or you can use a completely unstained sample. I really like to run an unstained cell sample and an unstained bead sample and use those unstained samples as the negative to match my positives, but it depends on the method you want to approach. Either way is okay. So once I'm happy with all of these setups, I can scroll down here and make adjustments. I can view that matrix by clicking on the View Matrix button up here, but I think I'm going to show. Yeah, so drop-down lists allow you to remove parameters or choose a different set of parameters or reset the UI to the original state, right? So you can change if it makes an incorrect assumption that this is the positive sample for that color. You can make a change and choose a different sample, or you can remove the parameter from consideration here. There are a couple different options for calculating compensation in terms of the math. The default is to use a traditional algorithm where we gate positive and negative events and then calculate the spillovers. There is also a checkbox button for calculating spectral compensation, that is spectral unmixing math. If you collect more parameters than you stain for, then this option becomes available to you, and it'll use linear least squares to unmix the data as opposed to just calculating spillover coefficients with the traditional algorithm. And then if you check this button for AutoSpill, this AutoSpill algorithm actually removes the need to gate positive and negative and uses robust linear regression to draw a line between the positive and negative events and generate that spillover matrix that we use for compensation. It is a new option just in this newest version of FlowJo, so it's only been around for about 6-8 months, and it's getting a lot of attention because it, again, removes the need to do all of your gate adjustments and make sure that you have perfect positive and negative subsets here because it's not gated on that. I'll go into more details here at the end. So you bring in your data, put it into the compensation group, go ahead and click that comp button. It's going to auto gate, and then your job is to make sure that everything looks good. If you need to, as I said, double click, bring up a graph window, move the gate to a region where the expression exists. Sometimes, you know, if you're using CD14, it'll be up in the monocytes. You got to move the size gate to get those events. In this case, with the live dead stain, moving the size gate to the lower end of the forward scatter region will get me to some dead cells that then show up as a positive signal in this histogram. The green samples on the right are the positive stain controls, size gated, and then those size gated events show up as the green histogram trace. We're actually using the range gated green histogram fraction as the positive, and then the blue samples in the left are my universal negatives for cells and beads, and those are shown here in shaded blue histograms in the overlay in the middle. So I'm using the shaded blue as the negative and the range gated green as the positive for each sample. Scroll through, make sure those negative and positive pops good, and again, then you're ready to actually view the matrix and create the matrix calculation and apply it as long as you've accurately defined your positive and negative fractions. Once you've done that, you're happy with your positive and negative setup, all this window looks good. Viewing the matrix is the easiest thing to do before applying it, and I recommend you do that. You can apply the matrix to a group, but it's always worthwhile bringing up this window and making sure that these numbers look realistic. The more red shade or highlighting the cell it receives, the more interaction there is, so the larger compensation values. And what you want to look out for is negative numbers. If you mismatch beads and cells as a positive and negative with bad fluorescence, you know, if you use a cell positive and a bead negative, you can get weird numbers here, especially big blue negative numbers are probably meaning that you did something incorrect here in the setup and that you need to go back and reevaluate that. I would expect mostly positive numbers, and you know, fundamentally values over 100 are acceptable, especially on some instruments. You can't get away from it, but if you're doing any type of low or mid level, then mid parameter experiment, then you can probably set your voltages so that each comp is brightest in its own channel and all the spillover values will be less than 100. But fundamentally, there's nothing incorrect about having spillovers greater than 100%. It just can be avoided with better instrument setup. I'm sure that you guys can talk to the tech support team at BD and come up with a solution there. So this preview window, again, you can preview here, and we can drag this M button to the workspace to apply any of the matrices that we create. That's what this is showing, right? Open up this preview window, select the matrix you want to apply, drag this M button to the group, in this case all samples, and all of those samples will get the red badge showing that they're compensated now with my new compensation that I've just created within FlowJo, and instead of using the acquisition defined matrix. That's how you apply, drag and drop. Now this is the matrix for compensation, right? That's correcting the spillover of emission into different non-primary detectors. There is also a way to assess the spillover spreading, which is a different measurement, It's calculated using the same positive and negative gates traditionally that you created to create your compensation matrix, but a spillover spreading matrix is a special type of matrix. It's telling you how much loss in sensitivity you will see if you use this color combination, right? And what's interesting is that even though spillover spreading is related to compensation values, they aren't the same. The highest compensation interaction I had was PE Texas Red into PE Sci 5 right here, but the highest spreading value is APCH7 into PE Sci 7. So that's the color combination that I may want to avoid having bright signal here and trying to read out a dim signal there. The spillover spreading matrix can be accessed by just clicking on this SSM button and asking to display the SSM, or to export it will create a CSV file that you can then open in Excel and utilize as an assistant for panel building. What spreading has done is given us a way to use compensation controls to understand the reduction in sensitivity that we get from certain color combinations. And the big red numbers are the interactions that I want to avoid here. So large numbers with red shading show me that if I stain with the color in this channel and then try and read out in this channel, I'm going to have a hard time seeing dim reagents on that second, because this color is going to cause spreading of the population in this channel. And at the ends of these rows are the sums of all of the spreading coefficients for that reagent across all of the detectors here. And you can see some of these are really low, like 8, and some of them are really high, like 81. And so if you start out on your instrument by creating a spreading matrix, by running comp controls, and then generating this compensation, visualizing the spreading, you can actually use this table to inform your decision of color combinations that are going to work out well and which ones you want to avoid. And the first thing you can do is go, well, what are the colors that are causing the most spreading across the most channels? And that's the sum number here, and eliminating those. If I'm going to do a 20 color experiment, I've got 8 dispensable channels on this symphony. I can drop those channels that have the most, that cause the most problems with spreading, and therefore create a panel that has nice sensitivity. I'm sure that the applications team can help you build those panels and can give you options for reagents that have less spillover interaction if you find that you have a bad spillover problem. This compensation control here shows that spillover, right? We have compensation that's been corrected for by bringing that skewed population down and making the medians of the positive and the negative equal. But now I have this widening or spreading effect, which means that if I stain with my CD3 reagent, I'm not going to want to use APCH7 on a really dim marker in the CD3 background because I'm losing sensitivity here. Instead, I might want to use APCH7 for a completely different lineage, or to use APCH7 on something that's going to be very bright so I see a good double positive signal. So again, anytime you have these spillover spreading coefficients that are very high, you may want to avoid, it can inform you on how to avoid color combinations that cause reduction in sensitivity of your readouts. And it's become a real essential tool to building really nice, high resolution, high parameter panels. You can always edit your compensation after you have created it, but you can't edit the original first compensation matrix you create. That one is static, just like the acquisition defined matrix. You can't go in and select these numbers and change them. You can click this edit button right here. It's actually not on the right spot there. The edit button allows you to create a copy of your matrix, copy, and then those copies are editable. So I can always go in and change the values here. And again, if I'm changing values, I will probably apply this compensation to my comp control and ask the specific question: how is this fluorochrome reagent bleeding into this channel, and is it being corrected correctly by using this medians are equal in the spillover channel type of approach? And I'll show that to you. Save your compensation. That can be done through this Save Matrix button. If you want to bring your compensation back into another FlowJo workspace, save it as a matrix file, which has a header information FlowJo XML so it can read, the workspace can read it, or you can save as a CSV file, which is just the raw matrix. You can open that up in Excel and heat map it and display it or share it. Click the plus button down here. You can navigate to that .mtx matrix file, and then it'll come in as an actual matrix that you can select and apply when you're ready. So, having said that, let me just jump out of here. I'm going to run through that real quick, and then we're going to switch gears once more to plugins. Here's my compensation group, right? It does not have compensation applied, but I can double click and I can see my acquisition defined compensation, which is actually applied to all the other samples except for the comp controls, right? So the lattice work here is filled in. When I get there, I can take that matrix and I can choose any preview sample, right? I can apply to a sample that's actually been stained. I can go down to a gated population. And when you zoom in on an individual color combination, this is Alexa 700 bleeding into APCH7. You can see the blue is what's overlaid, the raw data, uncompensated data in blue, overlaid onto the compensated data in black. There's a checkbox to overlay that, or you can remove the overlay. And what I'm looking for here is nice, straight, square, biological looking populations. If you see skewing on or off the graph into the middle or into to the edge, that means it's under or overcompensated. Visually, you can detect that quite often just by looking at the compensated data itself and comparing it to the raw data. So just know you can preview all the color interactions here, uncompensated versus compensated, on any sample at any level in the gating hierarchy. Again, if I want to edit this matrix, I can click this edit button. It will make a copy. And now those interactions are editable. So look what happens when I grab this 35 number, which is right here, and I reduce it to 25. Press return. Notice that black population, the grays, just started skewing in towards the center. It's no longer square with my negative, and it's kind of following the line of the uncompensated data, indicating that I have undercompensated this interaction between those two colors. Going the other way to 45, notice it's skewing off the edge, right? That looks very unbiological. It does not look square. It is an overcompensated interaction. And as you get used to looking at these, you will notice that in the preview and be able to focus in on that and ask, why is that not compensated? Maybe it's not the right compensation control. Maybe it's a mismatch. Again, good compensation comes from having good single color controls in the first place. And then I can go back to my original value, which is around 35. And there it looks nice, like I would expect it, that interaction. So that's editing a matrix. You can go in and you can change these numbers. If I want to create a new matrix, I don't like the acquisition defined, or maybe you never applied compensation when you collected the data, so that it comes through the FCS doesn't have a matrix with it. Well, then that's where we go to our compensation group, right? Compensation group has all my comp controls in it. I hit this this compensation button up in the static toolbar, easiest way to get to it, and it's going to auto gate my samples with a size gate and a range gate for each sample, giving a positive fraction of events that has that fluorochrome and therefore the emission spectra isolated on it. It brings those samples into this preview window, and now I can interact and make my choices. So I have a blank channel that was open but not stained with any reagent. I'm not going to include that channel in the compensation calculation, so I can go ahead and click the drop-down sample list and remove this parameter from the compensation. That goes away. So I'm down to 9 parameters in this experiment. And then I immediately start going through each channel. You can click on it here to get there, or you can slide the slider bar and go through each one. What we're looking for is nice positive, nice negative, and I immediately have a problem here. So I've got my unstained cells in blue, my stained cells in green, and it looks like it just gated on the negative. So why is that? Because my dead cells, I've said this before, but they're lower on forward scatter. So double click on that population, get on that preview, and then I move that gate down to where I get some dead debris stuff. And look, there's now my positive population there. I can go into that. I'll just do it here because it brings it up with the right parameters showing. And I can move that gate to define this as the positive region, right? So there's one second sample is a cell, is a bead comp, and it looks really nice, positive and negative, gated appropriately, beads. Another cell comp, this time for my CD3 reagent, but it's nicely gated and separable, so no problem. And that's it. That's all I have to do. This is just a transformation. I actually have stained the CD38 so high that my removal of a half a decade of range up here has cut it off, but it's still there, so I'm not going to bother with going and changing that transform. Nice and bright, right? Interferon gamma, nice bright signal. You would never get that with a cell without actually stimulating the cell. So again, beads are easy to use if you have intracellular markers, and just generally easy to gate. You know you've got a nice positive signal when you're using beads. So my compensation controls are good, and it was really easy for me to auto gate them because they had nice bright peaks. The only problem I have is having to move this size gate to accurately define the dead ARD positive events within this cell sample. I'm done. I'm ready to move on and view the matrix. Traditional matrix will create slopes between the positive and the negative for each channel and calculate those coefficients. So viewing the matrix here, it calculates the spreading, it calculates the compensation. The computer's really quick at inverting that spillover matrix and giving us the compensation matrix, and you can see the numbers are almost identical to what came off of the instrument. If you actually want to compare two compensation matrices, you can double select them and it'll show the differences. This is the deltas here, right? And so they're virtually identical with 0.006 difference. And then it'll show the two colors here overlaid onto each other, and you can see that the events are virtually identically on top of each other. So in this case, my calculated compensation in FlowJo versus my calculated compensation on the instrument match almost identically. I can use either. I'll grab the M button, selecting this compensation matrix, and drag and drop it to my samples, all samples, and now I have these badges turned red, including my compensation. And so when I go back and I want to, verify that any interaction is perfectly compensated between one fluorochrome bleeding into a secondary spillover channel, I often like to go back to my comp controls here and do that statistic calculation, which is I'm going to look at my size-gated Alexa 700 stained cells and look at Alexa 700, right? So there's my positive population, my negative population. In the spillover channel, HLA-DR, and what we can do here is gate for neg and positive subsets on the single color comp control. I can even visually display medians here as these boxes. And then what I'll do is take these gated populations and just drop a statistic on them, which is the median in the spillover channel. So if you have accurate compensation, these numbers will match or be very, very close, which they are within a point. I'm totally satisfied with that. If they are off, that is, the compensation is not correct, so I'll illustrate that real quick. I'm going to grab this acquisition defined copy, drag it onto my samples. So now I'm applying the blue matrix, and then I'll go ahead and change the matrix value from 35 to 25, right? And look what happens. The gate doesn't even move, and neither does that update, but it skews in just like we showed. And now look at these numbers down here for that sample. Median of the negative is still 5, and the median of the positive is now 160. That is completely off. It is poorly compensated. You can not only see it skewed visually, but you can see it in the medians of the two populations. And so when I come and I start to mess with a matrix, I often will recommend you to use a visual dot plot as well and gate on positive and negative and make that statistical calculation and try and match the medians on your comp control right here. So there, I hit it perfectly with my 35. I knew 35 was the right number, right? 2.9 to 2.9. That's a correct compensation interaction. So you can imagine that when you're calculating compensation, all the work is actually in this setup, right? If you have good compensation controls, nice clean controls with clearly defined positive and negative peaks, it's really easy for the algorithm to auto gate those peaks and you barely have to do anything. If you have kind of messier controls or not normal controls, like you're looking at antigens that aren't on the major population of events here, or you're looking at an antigen or an expression of a fluorochrome that's going to be not within your automatic size gated region, you may need to double click and make a move of this gate to find the expression, right? CD14 is going to be up here, not down here, and you may need to move that gate to get it. But those are the type of adjustments you need to make. Auto gating usually does the trick, but if you need to, you can always double click and make a change here to the gating and move that as necessary or widen it out. Now, that is the basics of compensation. Define your populations, calculate by viewing the matrix, and then apply by dragging the selected matrix to a group of samples, applying that matrix calculation to them. But as I said, we have a few different ways of calculating compensation now, and you can choose those different methods by using these radio buttons for traditional compensation, which is what we've just done, versus AutoSpill, which uses linear regression and removes the gates, versus spectral, which requires you to have more channels open than actual stains in the system for spectral to work. So again, there are spectral instruments out there, but any instrument is technically spectral as long as you collect more channels than you stain for. Now, I'm going to go back to the presentation here, but just know that AutoSpill is what I'm going to focus on for a few minutes. And what AutoSpill does is it actually removes the need to gate positive and negative peaks and will automatically calculate compensation based on linear regression of the events instead of the positive and negative fractions. And so I can view the matrix and use AutoSpill and get a spillover spreading matrix and a calculation of the compensation in a way that removes my need to go through and make sure that these positive peaks are all gated accurately. So let's jump back here and I'll do it real quick. So cleanup gating obviously is essential. You may acquire on a log scale for side scatter, and it makes it a little harder. Some clinical groups really like log scale. I personally would prefer this type of approach where you clearly get separation of the grands, monocytes, and lymphocytes and debris by using a linear scale for scatter. But either way, you can manually gate or let FlowJo auto gate the size gated or the major population in there. And what you're looking for is to find or identify cells with that consistent autofluorescence and focus on the population that expresses the antigen or is stained with the fluorescent reagent you're interested in. Now, traditional compensation, like I just showed you, is all based on you gating for positive and gating for negative. This is usually done on a histogram with our user interface, but you could also just as easily gate in a two-dimensional plot, which will show you the actual spillover. So here's another example of one detector spilling into another detector. So the emission of this fluorochrome that's being picked up in detector 1 is spilling into detector 2, causing this population to look double positive in the uncompensated state. We have to gate positive and negative, and then we take the medians in both channels and we draw a line through those medians and actually correct for that spillover. So spillover calculations are all about traditionally defining the correct positive and negative, drawing a slope through the medians and compare this detector, this dye, and see how it spills over into every other open detector and make that coefficient calculation based on a slope of this line. Right? So we have to draw through the medians a slope and get a slope to calculate traditional compensation and fix it. So that's easy, right? Just gate positive and negative. The problem is it's not that easy on a lot of samples to gate positive and negative. You have samples with low frequency of expression, low antigen density, or even autofluorescence that can compound issues here. So this is one example of that, TCRV delta 1 in FITSI, it has very, very low levels of expression. And this is the way it would gate automatically in your FlowJo, in your FlowJo compensation wizard. But FlowJo also requires 2% positivity to reach a certain threshold there for saying it's a good compensation control. And so when you have very low antigen density, if you look at a different color here on the Y, you can see that this is spilling into this. FITSI is spilling into the BB630 channel here, and the actual median that we would be drawing a slope to and through is really low. It's not way up here, it's not an easy one. It's very shallow slope. And so my question to everybody is moving the gate better? Does that actually fix this low frequency expression, low antigen density sample, and make a better comp control? And the answer is really no. By moving the gate, I've actually moved the median point for all of these gated events even closer to the negative, and may have an incorrect slope when we draw it through those two medians. And so just making having a poor expression compensation control is a problem that with traditional compensation we weren't able to fix, because we were using gated populations and drawing slopes through the medians instead of using linear regression and considering all of these events out here and finding a slope of a line that fits that. So AutoSpill, as I've mentioned, is an alternative method to traditional compensation, where it takes those positive and negative events, it takes the entire sample and it calculates a robust linear regression, linear fit line in order to get the slope calculation. This means that I don't have to actually gate for the positive and negative, which is really helpful when you start working with 20, 30 colors at a time, and you don't have to go through each one of those lines and make sure that the gates are positive and negative. It's also very helpful when you're using cell compensation controls that have lower levels of expression, or just a skewing, a low antigen density tertiary expression level. So we still use these single stain controls. You put your compensation controls into the compensation group, and you still calculate the spillover coefficients using the slope of this linear fit line. You just don't need the positive negative gates, fundamentally simplifying compensation. From the paper, this shows robust linear regression. AutoSpill really works similarly to traditional compensation. Robust linear regression means we're weighting the points that are far away from the line less, so you get a better fit. And you can see in this first column, traditional compensation corrects with the blue is the uncompensated data, the black is the compensated data. They're just showing two examples of bleed into a channel that is corrected. If they run the AutoSpill algorithm using the robust linear regression, just the first round of that linear regression actually recapitulates the traditional compensation. And you see this skewing because we're on a log scale here. If you actually convert the axes into a linear scale, you see the linear relationships of these events as they go up in signal. It's just an artifact, the skewing or the shape of the curve here. When we look at real data, that it looks like it's like arcing. That's due to this bi-X transform and being on a log scale here, not as soon as you change that to a linear scale, you get a nice straight line. And so it really works, and it's impressive at how well it works, especially for cell compensation controls that have an internal negative, and you're just running, creating a linear fit to that data. So AutoSpill doesn't just compensate once, like traditional compensation using a single line, but it actually iterates. So it compensates, and then it refits the data and estimates error, calculates a new fit, and it repeats this process until it converges on a solution. And that's really helpful to get the best possible compensation. It means that the chances of you having to go and manually edit the compensation are lower using AutoSpill. The other benefit of AutoSpill is you can actually generate an autofluorescence vector using a biological sample that shows autofluorescence of cells. So you can actually remove autofluorescence using AutoSpill by opening up another channel, not staining for it, and then just compensating that channel using the unstained cells as your compensation control. And in this case, this is a real example. Unstained mouse brain cells. If you look at the UV into blue, U8 into blue 2, you see a nice autofluorescence profile of a subset of cells within this mouse brain slurry or single cell suspension, and you can actually calculate a robust linear regression line and correct for that, subtracting out the autofluorescence of these cells from your system. And traditionally, you want to choose a parameter with highest autofluorescence, and oftentimes, we find that the UV Now, it totally depends on you. You want to run unstained mouse cells and see, or unstained cells of the cell type you're interested in, and see which one actually shows autofluorescence in the channels that you're collecting, and choose that as the as an additional channel to open up on your instrument. Now, I'm just going to blow through this, but we also create a spillover spreading matrix with AutoSpill, but it takes a slightly different method, partitioning the data, estimating fluorescence in primary and secondary detectors, and calculating a linear fit. Then we like shift the line, adjust the data, and calculate a linear fit through the 0.2 to determine the spillover spreading coefficients. Because traditional spillover spreading requires that positive and negative subsetting, we've come up with a way to generate spillover spreading matrices without those positive negative gates. And in most cases, this recapitulates a traditional spillover spreading matrix that is virtually identical to that traditional algorithm. So to conclude, we have a new compensation method called AutoSpill, and it requires less adjustment of gates on controls and works quite well with biological cell compensation controls to generate compensation matrices that should not require manual tuning of the matrix because of this iterative nature. The other huge benefit of AutoSpill is that you can subtract autofluorescence with an extra channel. And again, this works best when the channel has a high level of signal in the unstained control. So you want to sacrifice or choose the most autofluorescent channel when you design your panel to subtract out autofluorescence using an unstained control and enhance the resolution of all your other channels by removing that autofluorescence. Now, the limitations here is that really this AutoSpill algorithm works best when you're using creating linear regression lines on cells in a single tube. Now, that doesn't mean you can't try it on beads and use beads perfectly fine with it, but the initiation of this algorithm was really to come up with a solution to those low-level expression or those poorly stained samples that don't have a great positive and negative, clearly defined positive population, and that mostly arises on cell compensation controls. So usually you want to have an in-tube negative and just one sample as the positive and negative together. This iterative nature means that sometimes data sets fail to converge on a solution, and they point this out in the original paper. I've seen it once or twice where AutoSpill will not find the right solution, and in that case, you want to revert back to traditional compensation. But it is worth comparing to traditional if you want to, eliminate that step of gating on all your positives. And most clearly it's important to subtract autofluorescence on certain cell samples. It can have a drastic effect on the true readout of removing autofluorescence, can give you cleaner data, but you can't measure autofluorescence on a bead, right? It has to be a cell, and you use unstained cells of the type that you're analyzing to remove the autofluorescence. You can't use bead comps for that purpose. Another example from the paper, if you don't compensate correctly, you can get total wrong gating and shifts. In this case, the poor compensation led to the gate not having the events of interest in it, and so you end up with much lower than expected frequencies. And this was due to an incorrect gating in traditional compensation and poor compensation controls. The moment they started using AutoSpill, they get proper compensation and the correct enumeration of the frequency downstream. And so this is a new tool that has been out for only about 8 months, since last summer. I encourage everybody to try AutoSpill just by checking the radio button there in the user interface and calculating a compensation that is not using positive and negative gates. And all you have to do here to do that is click the AutoSpill, and then I would go through and I would actually drop all of these guys and only use, I remove all of the negatives and only use the in-tube positive and negative of each of my samples to calculate the matrix. Click the View Matrix button. It'll show you a iterative process as it goes through and give you a compensation on the end. Okay, so Amlan, I am done with this compensation spiel, and I hope that was helpful. Are there any immediate questions that I can address, and then I will go into a quick intro to plugins setup for you guys.
I think for the sake of time, we can have like the live poll right now on compensation, and then we can directly go to the plugin setup.
Sounds good.
Sure, Amlan. I'm launching the first question. It's there for you, Amlan.
Okay. So it tells PE Psi5 as a fluorochrome can spill into the detector meant for PE fluorochrome. So the options is true because PE Psi5 has a tendency to exhibit two photon phenomena, sending emission in lower wavelength. Second option is true because PE Psi5 is a tandem for PE and there can be residual emission. Third is false because spillover only happens towards the higher wavelength and PE detector is at lower wavelength. And last option is false again because PE Psi5 does not exhibit spillover characteristics. So please go ahead and select the right answer.
Amlan, we are seeing B as the option which most of the participants are selecting. 16 responses for the same.
Okay, looks good. Okay, all of them are responding to B, which is fantastic. The correct answer is B. So shall we go to the next one?
Yes, Amlan. So the second question is like,
okay, I can see it right on my screen. Which of the following describes the relationship between compensation matrix and spillover matrix? The first answer is single color controls gives us spillover matrix but never the compensation matrix. Okay, option B is compensation only works for up to 18 colors. Anything more than that requires spillover. Okay, and C is compensation and spillover matrix works up to 18 colors. Anything more requires AutoSpill. And D is the inverse of spillover matrix gives us the compensation matrix. Okay, we can have the answers now. Participants, please select the answer which you feel is correct.
we are seeing responses towards D and now one towards A. Yeah. Mostly everyone is pointing towards D.
Okay, that's the right answer. The inverse of spillover is the compensation matrix, and we can see majority going with that. Great response.
Yeah.
Okay, I think we can have the next question.
It's live now.
Okay, why is cleanup gate important in compensation? Option A, it can help to remove false positive signal. Option B, it can help to remove false negative signal, example autofluorescent cells. Option C, it can help narrow down to the cells that have the right expression for the probe interrogated. Option D, all of the above are correct. And last option is none of the above are correct. So please go for the answer. So we are getting answers. Most people says it's D.
All of the above are correct. Yeah, we're seeing that as the response.
Some options are coming also like C and
From
B.
B.
Some.
Yeah. Yeah.
Okay.
But
Okay.
90%
So
said,
yeah, more than
the D.
90% says option D is correct, and that is the right answer. So we can have the fourth and the last question for the session.
It's live now, Amlan. You can read it.
Okay, what is the one advantage of AutoSpill over traditional compensation? A, it is extremely fast, faster than traditional compensation. B, it is possible to perform autofluorescence extraction provided an open channel is provided. C, it is less demanding on gating. And D, both B and C are correct. So I'm so sorry. We have one more option. A, B, and C all are correct. A, B, and C. I didn't see that. Sorry. We can see different responses here. Most of them are saying option E, which is A, B, and C are correct. Some of are saying B and C are correct, and others are 0. Okay. So the correct answer to this is both B and C are correct. Okay.
I think, Amlan, you can tell them, I mean, why A was not correct. So, because most of them said A, B, C are correct.
Yeah, Tim, would you like to take that?
Right, so definitely less demanding gate adjustments and autofluorescence subtraction are the answers there. In this case, AutoSpill is actually slower than the original traditional matrix calculation because of this iterative computing. It's virtually instantaneous for like 10, 15 colors, but when you get up to 24, 28, the iteration process of calculating these robust linear regression lines takes a little time, and it'll show it'll take longer than the traditional approach to calculate, and you'll see a progress bar while it does, but it could take, I mean, on a 28 color matrix, it could take a minute or 3 to actually come to the final solution there.
Great, thank you.
Yeah, thank you guys so much. I know I've gone a little slow here. It's a lot of content to cover, and we've taken our little breaks and asked some questions here. So this last section is all about setting you up for next week and the week after to give you an idea of how to set up your tools for some additional functionality within FlowJo. These tools that I'm mentioning, we generally call plugins because they are external little files that you can add to your FlowJo setup and to run an algorithm or a process that isn't within the base package of FlowJo. So I just want to go through what fundamentally are these plugins, where do I get my plugins, how do I set up the plugins, and why you might want to use the plugins. And really this last question, we're going to address the why and the how next week with our sessions in more detail and show you some workflows of how we could employ these plugins sequentially as parts of our analyses for high parameter data analysis workflows. So as I said, plugs are apps that you can download and add to your FlowJo setup to run an external algorithm or function outside of the base package. Some of these plugins can be used to clean up your samples automatically, go through and clean up bad events, remove them, embed data in fewer dimensions that we call embedding, dimensionality reduction. We can use them to classify events into populations based on their similar phenotypic properties, that's clustering, and we can use them to visualize population relationships or make comparisons between populations. But importantly, you have to download these plugins and install them before you can use them. The plugins menu within FlowJo is located under the workspace tab. There's a little plugins icon, looks like a puzzle piece, and if you click on that, you'll notice this drop-down list appear. If you've just downloaded FlowJo and you've never used a plugin before, this list will be relatively blank. It'll just have a few items at the beginning. And the first line item is the FlowJo Exchange. That is a link to actually links you out to our website where you can download these plugins and the information on how to set them up and install. Once you have plugins installed, when you click this plugins button, you'll see all these additional options show up in the drop-down list, and you can point to data and you can initialize from this list a function in a plugin form, just like you can click a button within the ribbon of the workspace. And so the Exchange first line item there will take you to our website, and you'll see a whole list of all of these plugins. If you click on the plugin, you can get more information, and it has previous versions. The most recent version can be downloaded by clicking the arrow button. It tells you whether this plugin is good or can be utilized within the FlowJo application or the SeatGeek application for single-cell multi-omics analysis. And there's a number of different types of plugins, right? You'll see FlowSum and UMAP and Phenograph and Cluster Explorer. These are clustering algorithms, dimensionality reduction tools, and even user interface where you can mine that data and make comparisons. And so again, click on the download button to download any plugin you're interested in, and that downloaded plugin will come with associated documentation. When you install your plugins, you have to go through this process the first time you use a plugin. Once it's installed, you can just point and click to it, right? But there's several different types of plugins. Plugins that run just by themselves, and then in that case, all you need to do is download the plugin and put the plugin jar file, that's the jar for Java file, in a plugins folder, which is on a PC, it's going to be within your FlowJo applications folder. You will find a folder called plugins. You put that jar file in there, you restart FlowJo, and then that plugin will become part of your list. It'll show up in that drop-down list. If you've never used a plugin, you do want to go to your preferences and set up the configure tab. I'll go through that. Show FlowJo where your plugins folder is. If you move it, you need to say, my plugins folder is now located in this direction. And you can show where your R installation is. And R is a statistical computing environment, a programming language for data analysis. And we often use plugins to call out to this R installation and run an algorithm that is written in R code and then return back the results to FlowJo. So if you've never used R before, you'll have to go and install R, and then we have to run a plugin package installation script to download the proper tools within R to run the actual plugin process, the algorithm. And so to break that down for you quickly, we first download a plugin from the Exchange, and it comes down to my downloads folder here. It'll come as a zip folder, and you can unzip the folder and you'll find a few items. There'll be a jar file right there. FlowSum's open source. It's Sophie Van Gassen, so there's actually the source code in there if you want to get at how to create a plugin. That's a good example. Oops. And then there's a how-to document. And if you open that how-to document, it'll tell you whether this plugin requires R or doesn't, and if it does require R, it'll give you the installation script that you need to run within R to install the packages that or dependencies for this plugin to run and return a result. So the fundamental thing is to download, unzip, and then grab the jar file and put it into your plugins folder. In my case, I'm on a Mac, so it's applications. In my plugins folder within my applications folder, I have all of these jar files that show up in my drop-down menu. Again, restart FlowJo, and those plugins that are in your plugins folder will show up as line items in that plugins list. To configure your diagnostics preferences, right, to show FlowJo where your plugins are and where your R installation exists so that we can call out to R and utilize R in the background to run some of these algorithms, you go to your heart icon within the workspace, diagnostics preferences, and then there's two lines that you need to fill in. You can choose, you can just navigate to the location of your plugins folder and choose it by using the choose button, or you can type in the file path to get there, and you can set your R path. This R path is it's going to be different depending on where you install R. On my Mac, it's going to be in my user account, in my local folder. There's a bin folder, and there's where my R installation is. If you never change the location of your plugins folder, you can actually leave this blank. FlowJo will automatically scan for plugins, look for your plugins folder in its default location, but you can move that somewhere else. And in that case, if you move the plugins folder to a different location, then you do need to specify where we find the plugins on your operating system. So I always just go ahead and set up both of these by choosing and navigating to the proper location. And again, a restart of FlowJo, when you make changes in preferences, will apply that change and save it to your preferences. Setting up R. It looks scary the first time, but it becomes really, really natural once you've looked at it a while. R is a programming language, right? It doesn't come with a graphical user interface like FlowJo where you can just point and click. You have to actually write code. And the easiest thing to do is navigate just searching for rcran on your browser, and the first hit is going to be the rcran project, right? This is a shared open source project for programmers to share their packages, and that's where we get these algorithms. A lot of bioinformaticians will write algorithms in R put them up on a repository and give them to people for free to use. But if you don't know how to code and use R, then you're kind of out of luck. So what we've done is create plugins so that we can utilize these tools without you actually having to run the code within R. And the basis for installation of R is to click on the link as appropriate for your operating system and then download the base package. In this case, I'll go to my Windows. For you guys are mostly going to be on Windows. You want to get the base package. That's what you get to install R for the first time. And I would also recommend you get the R tools package because certain setups require R tools to be installed, otherwise it won't set up. Certain plugins do require R tools. So I get both the base and the R tools on a Mac, or I'm sorry, on a PC. And if I'm using a Mac, I'm going to download the base package of R and then also get this Xquartz program, which is required for certain plugins if you're on a Mac. So there's actually two things to download and install to get R up and running: the base package and then either R tools for a PC or Xquartz for a Mac. Installing R packages, once you have R installed, it's just an application. You double click and it opens up a window. It looks like this. It's a console, right? And this is where you write code. And what we're going to do to install our plugin code is actually open up one of these how-to documents and copy and paste a set of scripts here, which will be clearly annotated in the how-to document. We copy and paste this install script, and it'll install these packages that are required, including FlowSum, FlowCore, FeatMap, and such for various visualizations and clustering. This is what it looks like in the console, and you only have to do this once the first time you use an R-based plugin, is install the packages necessary to run it within the R environment. After that, it's point and click from within FlowJo. So blue is what I've copied and pasted, and red is what it's downloading. And you just copy and paste the script. Occasionally it will ask you, do you want to update packages, especially if you already have R and you maybe have some older out-of-date packages. And I always recommend to say, A, yes, update all packages. And then occasionally it'll ask you to download something that requires compiling. And if you see that, do you want to install from source packages which require compilation? The answer is no, because if you don't know what compiling is, you probably shouldn't be doing it, and it's okay. It'll work just fine. And then once it stops and says, you know, you're done with your installation, you can close R completely. You don't need to save anything. But now you have the packages installed within your installation of R on your computer so that you can call out to R from within FlowJo and run this package, this algorithm, to get your downstream results, right? Which could be a returned set of derived parameters. It could be a visualization. It could be cleaning up data and giving you a new population downstream of your sample. And so don't be too scared. We have our applications team is here to help. My tech support team is here to help, and I will always be here to help as well. Don't be afraid to ask questions when you're new to this stuff. We have created a plugin wizard to try and simplify and assist with your setup process of your plugins. And so maybe the first plugin you download is the plugin wizard, and we start the plugin wizard just like you would any other plugin. Of course, you have to get the jar file into your plugins folder first before you can actually spin this up, but it has links to get R, and it automatically allows you to type in and set the paths. And you even have a button to look on the FlowJo Exchange here, and it'll compare your installed version of a plugin to the latest version that's available. And there's an option to get them all. And get them all is going to download all the plugins and try and auto-install them. There's some advanced options underneath here. I just want to warn everybody that this works if you have no issues with your connection, but like on my if you have an IT infrastructure that limits the way your computer talks to outside environment, it may not work. Get them all, or these links here to go and get individual versions may not work due to IT blocking the communication from our application through to the Exchange website. And so even on my own computer right here, I can't just click get them all. It blocks it and it won't allow me to do it. So I have to go to the website and download each one individually. But this is our first attempt at making it simpler for you to install R and get all the plugins set up so that you can use these without too much stress of going through these steps, which really look daunting, but they are not daunting at all. It's just the first time everything's new. These plugins come in all different shapes and sizes, varieties. So classifying them into groups is kind of hard, but you can think of them as being useful for different levels in your analysis. Pre-processing plugins like Cytonorm. This is a normalization across multiple to correct for batch effects if you do multiple runs. Very interesting plugin, also from Sophie Van Gassen's group. FlowAI and FlowClean are data cleanup. Dimensionality reduction algorithms create parameters and redistribute events that we can see in a single data space. Clustering algorithms create populations. There's various visualization plugins, some special plugins for various utilities like CBA and stain index. And then, my favorite one these days is Cluster Explorer, because what I'll do with high parameter data, and we're going to go through this next week in detail, is use an algorithm to clean up the data, then run a dimensionality reduction algorithm, run a clustering algorithm, and then view everything in Cluster Explorer, allowing me to look at the clusters within the dimensionally reduced data space that we create. And so these plugins really are there to make your life simpler. They take a little bit of setup, but it gives you a point-and-click interface that you can run data cleanup algorithms or do population discovery like we're going to go through next week. Explore and visualize your data in ways that the base FlowJo package doesn't allow natively. So just to tease you for next week, and Amlan's going to go into this in great detail next week, is employing plugins in a workflow for discovering populations. We're going to use plugins at the level for dimensionality reduction, and this is where I was mentioning earlier today about using heat mapping, the third parameter heat map, on a two-dimensional plot, like a T-SNE plot here, to show where the antigen expression is of particular markers and orient you with this new way to look at data. So these are all the events redistributed into two parameters in a map that we can then mine and explore. Clustering algorithms, we're going to use plugins to run clustering algorithms